Okay, it is now 10 o'clock a.m. Eastern Time. Welcome, everybody, to the Neurofeedback Intervention Development Opportunities and Challenges Workshop. This is presented by the National Institute of Mental Health, the Division of Translational Research. Just a few housekeeping notes and reminders for today. Participants have entered into listen-only mode, cameras off, and mics are muted. Please submit your questions via the Q&A box at any time during the webinar. Questions will be answered during the discussion sessions of the workshop. If you have any technical difficulties hearing or viewing the webinar, please note these in the Q&A box and our technicians will work to fix the problem. You can also send an email directly to me, Deborah Kratt at dkratt@mn-e.com. And with that, I will now pass it over to our workshop presenters, Dr. Christopher Serampoti and Michelle Hampson. Chris, it's all yours.
Thank you, Deborah. Good morning or good afternoon, wherever you are. And thank you very much for joining us today. My name is Christopher Serampoti. I'm a program officer in the Division of Translational Research at the National Institute of Mental Health. The mission of the National Institute of Mental Health is to transform the understanding and treatment of mental illness through basic and clinical research, paving the way for prevention, recovery, and cures. Our webinar was developed today with the goal of sharing the state of the science for neurofeedback intervention development for mental disorders, with an emphasis on real-time fMRI approaches to treatment. We've invited researchers and federal officials, and they've generously accepted, to highlight recent developments in early treatment research, efficacy and effectiveness trials, and regulatory issues relevant to the approval and implementation of device-based interventions. We have also asked panelists to share their thoughts on the challenges for neurofeedback research, as well as the opportunities that may lead to more effective treatments for mental disorders. I also want to introduce our co-chair, Dr. Michelle Hampson. Dr. Hampson is professor of radiology and biomedical imaging at the Yale University School of Medicine. Dr. Hampson and her group are focused on developing, validating, and applying novel functional magnetic resonance imaging approaches with a particular interest in the use of fMRI neurofeedback to treat and study neuropsychiatric disorders. Michelle, thank you very much for co-chairing this morning.
Thank you, Chris.
For today's webinar, we will have 3 sessions. Session 1 will discuss early stage studies of target acquisition and measurement and task development. Session 2 will present work on ongoing efficacy and effectiveness trials of neurofeedback approaches. And Session 3 will cover regulatory issues and the larger question of how neurofeedback interventions are translated to clinical practice. Following each set of presentations, we will have a discussion period moderated by NIMH staff. Audience members are welcome to submit questions to the Q&A box. Please note that we will be unable to answer any specific questions you might have about patients or grants or companies, but we can answer more general questions specifically and particularly those that relate to questions you may have about the talks. This is a public meeting. And the recording of this webinar will be available along with a summary of the meeting on the NIMH website. And as we get started, I want to save as much time as possible for our presenters and for the discussion section, so we'll get rolling. But I just want to thank every single one of our presenters, my colleagues at NIMH and Dr. Holly Lisenby for her support and encouragement to have this webinar today, and to thank you all for your time and for your good work. Okay, for our first session, we have 3 presenters. And they will be discussing the topic of early stage development. Dr. Stephen LeConte will lead us off. He is associate professor at the Fralin Biomedical Institute at Virginia Tech. Dr. Masaya Misaki is an associate investigator at the Laureate Institute for Brain Research. And then our third presenter will be Dr. Michelle Hampson from Yale University. So with that, Stephen, why don't you kick us off?
Okay, thank you very much. Can everyone hear me and see the screen? Well, I'm going to assume yes. I see a thumb floating up. So thank you very much, Chris and Michelle, and everyone that was involved with the organization of this. I'm really delighted to be here today to talk about early stage real-time fMRI neurofeedback and how to design those early stage experiments. And so it could go without saying, even for a general audience, that the heart of real-time fMRI is fMRI. And if we had more time, I think this point would come up more prevalently. But I would like for everyone to keep this in the back of their minds, because I think sometimes it's so obvious that it may actually be overlooked by us as investigators or by us as reviewers. And so we inherit all of the magic of bold fMRI, as well as all of the limitations of fMRI as well. Nevertheless, if we take this block diagram as kind of a typical fMRI experiment, we have an individual in the scanner who's generally guided by some stimulus-based task. And the fMRI is collecting a 3D movie of the brain in action. Now, of course, the heart of real-time fMRI is that during this data collection, we can extract signals through a variety of different methods, prominently region of interest, for example, or looking at functional connectivity across multiple regions of interest, or in my own work, trying to use machine learning models to decode what the sensory, behavioral, or psychological state of the individual is on an image by image basis or on an ongoing basis during the actual experiment. And regardless of what signals we're extracting, we can think about those signals as control signals. And for today's topic, those will be controlling generally neurofeedback types of interfaces. And I think you'll see a wide variety of those interfaces as the day goes on. More generally, though, just trying to weave in some plugs for basic science, these signals that we're extracting can really be control signals for virtually any component of the experiment that we might want to think about. And so really, as we think about the experimental flexibility that real-time fMRI gives us, again, we inherit all of the components of fMRI. So we need to think about field of view, resolution, sampling rate, but then we get a tremendous and I think novel degree of flexibility from this closed loop aspect of the experiment. And I'll try to highlight that as the slides go on. And then not neatly fitting into this block diagram, but of course there are many parameters in the pipeline, both before, during, and after a neurofeedback experiment that go all the way from, our inclusion-exclusion criteria to how we're instructing participants before they go in the scanner to exactly what we're trying to measure in terms of change and how we're trying to measure that change after a neurofeedback experiment. And so this kind of leads to the parenthetical subtitle of my talk, which is real-time fMRI neurofeedback has a lot of knobs. And I credit this quote to Sue Whitfield Gabrielli. A number of years ago, she just said this, and I instantly had this sort of flashback to myself as a college student trying to get an oscilloscope to give me the waveform that my circuit was supposed to be giving me. And of course, this is a fairly complicated interface for receiving just one or a few signals at a time, but actual control interfaces are massively complex when you're talking about things like aircraft carrier engine control rooms, or I think what comes to a lot of people's mind are things like the flight instrumentation on an airplane. And so this is my attempt to capture the daunting magnitude of the experimental flexibility that we have. And, this experimental flexibility, I think, promises great potential for future innovation. But this flexibility has also imposed a big challenge to us, a small, enthusiastic community, because in one way or another, we've all come to the realization that despite decades of neuroimaging data and even more decades of clinically relevant behavioral data, we're not yet ready or able to use first principles to design neurofeedback experiments. And so this struck me a number of years ago when I was designing a Q reactivity study with my collaborator, Pearl Chu. And a number of years ago, there were not as many substance use disorder real-time fMRI studies published, and we were really hard pressed to find unambiguous, compelling guidance from the literature. So for example, should we be using images or videos, and what types of images? Should we design a crave, don't crave experiment, or one that focused only on suppressing craving? Could we even think about jumping in blindly and trying to change craving at an early stage? And what regions of the brain would be most potent for changing craving, and so on and so forth. And so, of course, we're not alone. And it turns out that porting basic science for evidence-based medicine is a well-known issue in implementation science, and it extends well beyond our specific real-time fMRI setting. So here I'm just showing the cover from this report that talks about the quality chasm between scientific knowledge and the state of scientific knowledge and the quality of clinical care. And so what are some of the experimental parameters that are open to us as investigators? And, because of the limits of time, I won't be able to share all of them. I just kind of want to impress upon the fact that there are a lot of knobs. And as you go through today's talks, kind of pay attention to the variety and breadth and depth of applications within the neurofeedback results. And this is just kind of a small slice of what we're going to be able to talk about today. So one resource I ended up relying on a lot, I ended up throwing out my own bullet points and using a review article from 2020 by Samantha Fede when she was in Reza Mominen's lab. And of course, as a review article, it's going to be locked in time and missing several important recent studies. But they covered a lot of important topics, and I think this paper covers them very thoughtfully. And also, I like the graphics because it kind of reinforces this idea of knobs and dials. So I think I was sort of drawn back to this paper as I was thinking about this oscilloscope again. And so, one of the knobs are the types of tasks that we want to do in the scanner. And again, we have to do them in the scanner. And so we have to think about the environment, which is somewhat claustrophobic, highly magnetic, and also loud. And so it's possible to do auditory tasks. It's even possible to do a limited number of motor tasks, usually manual types of tasks. There's no dancing allowed in the scanner. But by and large, the feedback interface is probably going to be visual. You could do tactile as well. Anything is possible, but it takes some engineering challenges sometimes. And then the current state of the literature shows that most real-time fMRI studies to date have really been focused on healthy populations. And I'm going to tie this into kind of the concept of positive controls because a lot of these papers have been technology development or sort of intended as stepping stones to jump into other clinical populations. And I mentioned briefly that there are many algorithms for extracting signals of interest. And of course, if you're an fMRI researcher or neuroimaging researcher or just a data analyst in general, you know that there are lots and lots of algorithms that are available. But to date, really most of the tractable first hypotheses have come from regions of interest types of questions. And this has actually been very productive because now you can see we're starting to grow a literature that's much more catered to our needs of trying to understand what regions are really accessible to participants to be able to use neurofeedback and even look at relative signal strengths that we might expect, which would help for experimental planning and for planning statistical tests, for example. Another area is for these ROIs or for a classifier that's detecting crave, don't crave, do you want to go up and down, or do you want to just be modulating in a suppression direction or enhancement direction? And those depend on the scientific question and the clinical context of what you're doing as well. And then for the feedback display, there are a lot of things that we could talk about in terms of feedback. And I think I'll save some of that for kind of the later talks. I would love to talk more about feedback. And I think feedback timing is important, but one area that we may have missed in this discussion is that we can also control stimulus timing. Because we have real-time access to the brain, we might be able to control when we present stimuli. So it's at a time when the brain is most vulnerable or most resilient, for example. And then the issue of control groups is something that I'm going to go into a little bit more in this talk with the limited amount of time that I have. So in the face of this complexity, how do we move forward to take advantage of real-time fMRI's potential? And I think that there are at least 3 interrelated ways to do this. The first is to embrace the NIH's experimental medicine framework, especially the idea of target engagement. The second is to leverage the idea of positive scientific controls. And the third is to utilize and remember that real-time fMRI is really useful as a basic science platform. And that goes hand in hand with our efforts to look at neurofeedback for clinical populations. So the NIH's experimental medicine framework is more than just about target engagement, but I would like to talk about the importance of target engagement through kind of an example. And in the interest of time, I'm not going to go through this example carefully. I'm happy to backtrack during the discussion. But essentially what it calls us to do is go beyond the traditional efficacy testing and instead have, in addition, hypotheses about mechanisms of action. And so that then becomes the target for us to test. And we can kind of reframe our questions with an intermediate target that's mechanistic in its nature in mind. And so then the issue becomes, how do we measure the target? And the great thing about real-time fMRI is that we can think about using fMRI as either, oh, no, I apologize, my slides for some reason have paused. Okay, we can use fMRI as a target and or as an assay. We could actually be using a target for feedback and assaying changes in parallel even. And so again, just to highlight on some of the experimental flexibility. I'm not going to read all of this about experimental medicine and target engagement, but I will say that a major part of the NIH's rationale is that studies that demonstrate target engagement are meant to be mechanistically informative with this really nice feature that the information should be there, whether the results are positive or negative. And so the other sort of motivation for this is that really the goal is to come up with generalizability and synergistic breakthroughs, recognizing that many unhealthy behaviors co-occur in different clinical populations, and they may have shared causal factors as well. Okay, so really quickly through positive controls, just as kind of a preview, and I think dovetailing with some of what Michelle will be talking about, she's going to talk about early, middle, and late phases of testing as kind of cast as a foraging exercise. And so in that early to late progression, we're going to be obtaining stronger and stronger evidence for both target engagement and clinical efficacy. And so we need to be more and more concerned about the specificity of neurofeedback in those results. And so that's where the idea of negative scientific controls, for example, the classic example is a placebo group comes into play. And so to talk about controls, a really useful article, again, I apologize, my slides are not advancing, so I'm probably going to extra advance, but I'll keep talking. So there's a terrific resource in a 2019 neuroimage paper by Bettina Sorger, Frank Skernowski, David Linden, Michelle Hampson, and Kim Young. And it's titled Control Freaks, but it's not too controlling and it's not too freaky, even though my slides are freaking me out here now. And so, I'll just kind of go through and eventually my slides will catch up to me. But essentially, the flip side of negative controls, which are testing for all the variables that could be confounding or kind of enhancing. So just going into the scanner might have an effect on participants, regardless of whether or not they're getting true feedback. And so you want to control for those with negative controls. Positive controls are kind of the safe launch pads in which to explore your next sort of experimental set of parameters. So positive controls are really useful. I think of them as sanity checks or debugging, but essentially you want some evidence that your system is working. And that could be as basic as whether or not your real-time setup is correct and all the computers are talking together, and or whether or not your task is actually appropriate or not for that. And so you may need to run control populations where you think that accessing the target might be easy or doable, and then move on to clinical populations where you hope to target the targets. So I apologize for both taking a while and, oh, now my slides are moving again. So just as kind of the final thing to say, in the face of this complexity, sorry, I really didn't do positive scientific controls justice, but the idea of using real-time fMRI as a basic science platform, I think, is a third thing that we should be doing. And so within the experimental medicine framework, we have to remember this ability to learn from successes and failures. And so the fMRI part of real-time fMRI, I think, demands post-acquisition analysis, including revisiting all of our instructions and trying to improve future targeting and mapping new potential targets. And, I'm not going to go through all of the creative experiments here, but real-time fMRI can sometimes get typecast as only a tool for neurofeedback, but this closed loop aspect really unlocks a whole new level of experimental flexibility. And so I'll just kind of end there. There's nothing profound in these conclusions, but I'll just leave them up and people can look back through the video as well. And I'd just like to acknowledge my lab and my previous funding. And there are many, many collaborators over the years, which, I deserve to thank. But for today, I'm just going to thank some of my active real-time fMRI collaborators. Thank you very much.
Thank you, Stephen, and thank you for your talk. We have for our next presenter, Dr. Masaya Masaki from the Laureate Institute of Brain Research. Dr. Masaki, feel free to share your slides and take it away. Masaya, you're muted.
So I'm sorry. So yeah. Okay, so I'm going to talk about discuss how we gain a mechanistic understanding of neurofeedback for precise interventions using real-time neuroimaging. So, okay, as Dr. Aconte has nicely introduced, neurofeedback involves many adjustable parameters, and there's no single best setting for them, as they largely depend on the specific context or goal of the interventions. So I believe that a deeper understanding of the mechanisms behind neurofeedback can help us identify effective parameter settings. So my talk will focus on the mechanistic understanding of neurofeedback treatment. So here's what I'll cover in today's talk. So first, I'll start with a brief introduction to real-time neuroimaging technology, focusing on fMRI. So this will build on the points from the previous presentation. And next, I'll move on to the mechanism of neurofeedback training. And finally, I will discuss intervention parameter settings based on mechanistic understanding. So real-time neuroimaging is a technology that analyzes and visualizes brain activity as it happens. So this enables a live view of ongoing brain activation. And it allows immediate feedback and interaction with ongoing cognitive process. So this technology allows for highly precise and personalized brain interventions, both in terms of location and timings. So with fMRI, we achieve spatial resolution down to a few millimeters, enabling us to pinpoint brain activation. And also immediate feedback and interaction can target the current or ongoing cognitive state. So this precision is expected to significantly enhance the effectiveness of the intervention. So neurofeedback is one of the most popular and promising applications of real-time neuroimaging. So in neurofeedback, we present brain activation signals to participants so they can learn to regulate them on their own. So while regulation is typically voluntary, but we can use rewards like monetary incentives for implicit reinforcement learning. And also the application of the real-time neuroimaging extends beyond neurofeedback. So we can combine this technique with various brain stimulation methods, such as TMS, TCS, FAST, or even with audiovisual stimulation. So by integrating these methods, we are able to fine-tune stimulation parameters while monitoring their effects in real time. So this integration allows for precise adjustments of parameters based on immediate feedback. So we can use a closed-loop iterative optimization of these other brain stimulation parameters. So for instance, in our proof of concept study, we successfully optimized TECS parameters using real-time fMRI, so demonstrating the technological feasibility of this approach. So real-time neuroimaging can also facilitate brain-based communication. So in this application, interaction partners such as clinicians can engage with participants while actively monitoring their cognitive or brain states. So we have implemented this system to demonstrate its potential for enhancing therapist-patient communications. So it's still a very early proof of concept stage, but their technological feasibility has been demonstrated. And also regarding recent technological advancements, the current real-time fMRI system enables comprehensive image processing of whole brain voxels comparable to offline analysis in real time. So for example, in our system is capable of applying comprehensive image processing, including a physiological noise collection in less than half a second. So in summary, real-time fMRI technology has significantly advanced, enabling precise real-time extraction of brain activation signals. However, it's important to acknowledge the challenges like the temporal delay in the board response and the limitations in statistical inference that still persist. And also several advanced applications of real-time fMRI are now technologically feasible. However, they still require high-level system integrations, and these systems often need in-house development and are not widely available. So this highlights the pressing need for more accessible and flexible real-time fMRI solutions. And despite these technological advancements, our understanding of how online stimulation or adjustment of brain activation affects cognitive functions remain limited. So in the next part of my talk, I will explore this issue further. So okay, let me recap the mechanism of neurofeedback training. So neurofeedback involves learning to control brain activation through self-regulation of mental states and reinforcement learning. So it's important to highlight that this process isn't just a focal intervention, even when targeting specific brain regions. So rather, it involves engaging large-scale brain networks through self-regulation, mental state monitoring, and reinforcement learning. So indeed, the involvement of large-scale brain networks has been demonstrated in several studies. For instance, a meta-analysis of neurofeedback studies, including various targeted brain regions, revealed commonly activated areas. So these regions are generally associated with skill learning and mental regulation tasks. So these activations are not limited to the neurofeedback training. So also our studies, which focused on increasing left amygdala activation in veterans with PTSD, have shown that the effect of the training extends beyond the targeted area. So we observed that DMPFC and MCC activations were associated with the training effect. And also the training effects were evident in the resting state function connectivity between the SMA and dorsal anterior cingulate cortex, were even in the hippocampus ranges. So these regions are distant from the target regions, left amygdala. Okay, given that the neurofeedback effects were not confined to the target region, it suggests that the impact on cognitive functions might be mediated through modulation in other brain areas. So this complexity indicates that establishing a simple causality between target modulation and treatment outcomes can be challenging. So for a more detailed discussion on this topic, I recommend to referring this paper. So can neurofeedback provide evidence of direct brain behavior causality? So it's a very nice paper for review. So at this point, it's crucial to consider that the appropriate outcome measure for training success. What is the appropriate measure of the training success? So should we solely focus on the regulation of the target brain signal, or should we also consider changes in cognitive functions or symptoms? So I believe that the primary goal of the brain intervention, including neurofeedback, should be to modulate cognitive function. So to achieve this, we select a target brain region that is believed to be linked to the desired cognitive function. And assuming a straightforward causal pathway where the target brain activation directly influences the target cognitive function, successful regulation in the target brain region could be serve as an intermediate measure of the training success. However, we do know that the neurofeedback training engages multiple brain regions beyond the target. And it may be the activation or network connections between these regions that actually drive changes in cognitive function. Therefore, successful regulation of the target brain region may not correlate directly with cognitive changes. So this means that regulation of the target brain signal alone cannot be considered a reliable measure of the training success. And also, it's important to note that even if this is the case, the target brain activation can still serve as an effective feedback signal for training if it reflects the brain activation that is causal to the cognitive functions. So however, if this is the case, we need to examine the whole brain process behind the observed cognitive change to fully understand the training mechanism and validate the effect of the intervention. So at this point, in this scenario, real-time fMRI is an ideal tool for investigating these mechanisms. So it not only supplies a neurofeedback signal for training self-regulation, but also offers a comprehensive view of whole brain activation during the training sessions. So real-time fMRI provides two variable outcomes from a single experiment, so making it a cost-effective approach for studies. So we should view real-time fMRI not only as a method for intervention, but also as a valuable tool for neuroscience research. So I'd like to share some of our recent findings from this line of research. So in one study, we did neurofeedback on the functional connectivity between the precuneus and right temporal parietal junctions. So this region's activation were positively correlated with the brooding rumination score in mood and anxiety participants. So we trained the participant with MDD to decrease this connectivity with neurofeedback. Then we observed that during the training, the connectivity between these regions indeed reduced, but there was no significant difference in this reduction between the active and sham control groups. So however, interestingly, we did find a significant difference in the changes in brooding rumination scores between these groups. So a statistically significant change in symptoms was observed only in the active group, not in the sham control group. So we conducted a regression analysis to correlate symptom changes with whole brain connectivity patterns using connectome-based predictive modeling. And we discovered that the interaction between connectivity patterns during the regression task and in response to the feedback signal was critical for predicting changes in symptoms. And furthermore, these predictive connections were not confined to the target brain region, but spanned across extensive brain networks. So these findings indicate that the symptom reduction was not solely explained by the regulation of the target brain region. And in another study, we conducted a whole brain analysis for neurofeedback study targeting the left amygdala So participants with MDD were trained to upregulate the left amygdala signal by recalling positive memories. So the details of this protocol will be presented in a later session by Dr. Yang. So I believe this approach is one of the most successful fMRI neurofeedback treatment for depressions. So I conducted the second analysis of these data set. And we observed a significant reduction in depressive symptoms in the active group, which was not seen in the control group. And additionally, the left amygdala signal was higher in the active group compared to the control group. However, the main effect of the group and group by line interaction was not significant. And furthermore, when we focused on individual variability, left amygdala activity during training did not explain individual differences in depressive symptoms. So we observed no significant associations between changes in MADRS scores and changes in left amygdala signal during training. So this suggests that the symptom relief may be mediated by brain activations other than those in the left amygdala. So here I assume that the variability in brain activation during the training could be linked to individual differences in therapeutic outcomes. So to identify variable brain activation patterns during training, we classified brain activation patterns into subtypes among active group participants. So this process involves the feature selection, dimension reduction, and clustering analysis, complemented by repeated cross-validation to find reproducible solutions. And we identified two subtypes of brain activation during the regulation block, so block activations. So the first subtype was characterized by activation in the control regions, like lateral prefrontal regions and supplementary motor areas. And the second type not only showed the activation in the lateral prefrontal regions, but accompanied by suppressive changes in the default mode network regions, such as the precuneus PCC and medial prefrontal cortex. And interestingly, significant symptom reduction was observed in participants categorized as type B, so who demonstrated suppression of the default mode network activation. And we also evaluated the event-related response to the feedback signal and identified three types of brain activation. In this case, we evaluated the brain correlation with the feedback signal amplitude, so event-related response. So type A showed a very limited response to the feedback signal. But type B brain activation showed a negative correlation with the feedback signal, and type C participants showed a positive correlation with the feedback signal. And again, significant difference in symptom reduction were observed between the subtypes, type B and type C, who showed brain activation that correlated in positive or negative way to the feedback signal, showed a significant symptom reduction 1 week after neurofeedback training. However, type A showed no significant response, showed no significant improvement. So we also noted a significant interaction between these subtypes, so it further explains the variability observed in the active group. And these maps illustrate specific pattern combinations within the active group that led to significant symptom reduction. So the presence of multiple effective combinations indicates that there are several potential pathways for treatment, suggesting that the route to depressive symptom relief is not limited to a single pathway. So in summary, the subtypes of brain activation patterns during neurofeedback training explain the variance in treatment effects. And the symptom reduction cannot be solely attributed to the successful regulation of the target region. And instead, the way in which participants regulate is critical to effectiveness of the treatment. Okay, so to conclude my presentation, let's consider how we might design interventions based on these observations. So when determining the appropriate outcome measure in neurofeedback studies, we should prioritize cognitive changes as these are the ultimate goal of the intervention. So however, regulation success can be considered a valid outcome if it closely relates to the targeted cognitive function. So, or if it is a part of a proof of concept study aimed at examining the controllability of certain brain activations and its effect on cognitive function, so this target is this outcome measure could also be valid. Okay, so our previous findings also raise a critical question about the optimal target for neurofeedback interventions. So should we focus on modulating a specific brain region, or it is more beneficial to consider large-scale activation patterns? So signals from a specific regional target are easier to extract, but often lack specificity for particular cognitive functions, and may require explicit and rigorous mental strategies to achieve the desired cognitive changes. And in contrast, large-scale activation patterns typically show higher specificity for cognitive functions, so potentially increasing the effectiveness of neurofeedback. And I would also like to emphasize that targeting detailed whole brain activation pattern is a unique advantage of the real-time fMRI neurofeedback. And that is difficult with other really focal stimulation methods. So however, linking these patterns to specific cognitive states requires the creation of a machine learning model, so which requires additional sessions to collect sufficient data for training. And the effectiveness of the intervention depends on the quality of such a decoding model. So regarding pattern-based neurofeedback, several innovative approaches have emerged. So including the Dr. Laconte's applications. So among these, decoded neurofeedback is particularly notable for its success. So this method delivers a neurofeedback based on decoded signal. So for cognitive or perceptual state that are derived from a matchboxer patterns. So typically, DECNEF employs a reinforcement learning approach, often using monetary incentives as rewards. And it also utilizes implicit training methods, which means participants are not given specific instructions on meta strategies. So another approach is connectome-based neurofeedback. So where the feedback signal is derived from the strength of thousands of functional connections instead of activation amplitude. So when connectivity patterns are a more reliable marker of cognitive states, so this method could prove more effective than traditional activation-based interventions. And also semantic neurofeedback is another innovative method that uses matchboxer patterns. So unlike decoded neurofeedback, this technique extracts the relative relationships between multiple cognitive states from these matchboxer patterns. So feedback is provided by presenting the representational similarity space between these states and the current state. So subjects are navigated to move their current state to the ideal state. So the selection of the target is also a challenge for neuroscience. So clarifying the brain activity associated with cognitive functions through ongoing neuroscience research will enhance the efficacy of neurofeedback interventions. So a significant advantage, so another advantage of real-time fMRI neurofeedback is its ability to immediately translate neuroscience knowledge into brain interventions. Thus, neurofeedback can evolve as neuroscience evolves. So there are many other parameters in neurofeedback, and including feedback timing as presented in the Dr. Laconte, and scan parameters and control conditions. But unfortunately, due to time constraint, I cannot cover everything today. So let me conclude my talk. So there is no one-size-fits-all solution for neurofeedback parameters, and utilizing a mechanistic understanding of neurofeedback training allows us to more effectively explore and optimize these parameters. And it's also critical to recognize that treatment effect cannot be attributed solely to successful regulation into target regions. So conducting whole brain analysis provides invaluable insight into the mechanism of neurofeedback training. Okay, so in closing, I'd like to express my gratitude to my colleagues, especially to my deepest thanks to the late L.G. Bojulka, who was instrumental in establishing our real-time fMRI systems and initiating our pioneering neurofeedback studies. Thank you for your attention. So, okay, I'll handing over to Dr. Michelle Hampson from the Yale School of Medicine.
Thank you, Messiah. And Michelle, we are a little ahead of schedule, so you have time, and we'll allow us more time for the discussion. So take it away.
Thank you, Chris. So I just want to start by thanking all of the staff at NIH who put all the work into organizing this workshop, and also to express just appreciation in general that NIMH supports these workshops, which I think are a great avenue for communication between program staff and extramural researchers. So thank you. So I'm going to be talking now about what I see as the promise and challenges in early stage development of fMRI neurofeedback interventions. So first of all, the positive, the promise. What I am most excited about with fMRI neurofeedback is the data suggesting that it really works in allowing us to do targeted alteration of mental function. So it's kind of a unique tool that gives us great flexibility for really intervening in humans and changing their mental function in a targeted manner. Many of you may be skeptical of that because biofeedback and neurofeedback have to some degree been seen as a sort of a hippie, not really evidence-based kind of stuff, but there is really pretty solid basic science literature now supporting this idea that fMRI neurofeedback is a tool that can enable targeted alteration of mental function. I'm talking here about controlled studies with participant blinding that have demonstrated changes in targeted aspects of mental function with large effect sizes. So just to give you an idea of what some of those are like, there was a study where they improved perception of a specific line orientation in subjects without affecting their perception of other line orientations. The subjects were completely blind to what was being trained, and they counterbalanced the line orientation trained across subjects, but they got very targeted effects. They've also shown that you can alter higher level aspects of cognition using this tool. A sort of creepy study was one where they basically trained people so as to manipulate their facial preference. They trained one group of people to like a neutrally ranked set of faces more, and another group of people to like the neutrally ranked faces less, and they got changes in the targeted direction. Again, people were blind to what was trained. So these are just some examples. There's actually quite a large basic science literature now that shows that fMRI neurofeedback can be successfully used to change mental function in people in a targeted manner, certain aspects of mental function, I should say. So just because you can change some aspects of mental function in a targeted manner does not mean you can do this with any aspect. And the critical question is, can we do this with aspects that are relevant for mental health? And I think you're going to hear a lot about the work that's going on in that front in this workshop today. And I think the early results have been quite promising, but it is still at an early stage. But I think that basic science research really gives us hope that this is really going to work potentially. The second thing I really love about fMRI neurofeedback is it's not just a maintenance therapy where you're going to have to keep getting neurofeedback regularly for the rest of your life to stay healthy, the way many pharmaceuticals work, for example. I think it has the potential to really shift people onto a healthier trajectory. So why do I think this? Well, studies that have looked at whether the effects induced by neurofeedback tend to persist after the training have indeed reported that they do seem to still be there months to years later. But even beyond that, we had a study where we were running neurofeedback in patients with OCD, and we were looking at whether the neurofeedback was improving their symptoms as measured by the Y-box, which is the gold standard clinical instrument for OCD. A decrease in the Y-box is an improvement in symptoms, and we were happy to see that during the period of time when the intervention was applied, symptoms improved in the subjects getting real neurofeedback and didn't change much in the subjects getting sham. But what we were really interested in was whether those effects persisted over time in the following weeks. At the 2-week mark, both groups showed improvement, probably because placebo effects were kicking in at this time point. Not surprisingly, as we went to the month time point, the sham group started to regress back to baseline, as expected for placebo effects. But we were a little bit surprised that the real neurofeedback group continued to improve. So we actually got quite substantial improvements in symptoms in the real group that occurred after the intervention had ended. And we were surprised at this at first. We thought maybe it's something idiosyncratic to OCD, because OCD is a disorder where if you resist your urges, that's a form of therapy for people. And so then they can get more confidence and start on a healthier trajectory. But we did have another study we were running at the same time in Tourette syndrome. And we thought, well, we'll just look in that data and see if we see any suggestion of the same pattern. And indeed, we saw exactly the same pattern. And around this time, I was going out to lunch with Nick Turk-Brown, who's another neurofeedback researcher at Yale, and I was telling him about these data. And his first reaction was, oh my goodness, we saw exactly the same pattern in an open label study of depression that we ran. We didn't know what to make of it, but if you go and get the figure from that paper, you'll see it's there. And sure enough, that's exactly what his figure showed. Symptoms were continuing to improve for weeks after the neurofeedback training had ended. So, then we started searching through the literature in general, and we saw suggestions of this sprinkled throughout. And so we wrote a paper about this, which was published in 2018 in Neuroimaging. The upshot of it is that it seems that changes in symptoms induced by neurofeedback not only persist after training, but in many cases continue to grow. And so this is really exciting from a clinical perspective, because it really seems like we're able to push people onto a positive trajectory that can lead to lifelong benefits. So the third thing I want to highlight about neurofeedback is that it seems to me to be a great intervention to pursue for treating youth. We're in the middle of this youth mental health crisis, and here we have an intervention which is safe and medication-free. It's based on natural learning, so it can leverage the brain plasticity in young brains. And also, as I just talked about, it has this potential to shift them onto a healthier trajectory, which in young people will lead to a lifetime of benefit. So for all these reasons, I think it's really a promising avenue for pediatric, especially youth, mental health development. And the last thing I want to highlight is what has already been really discussed by both Stephen and Messiah, is how beautifully fMRI neurofeedback contributes to an experimental therapeutics kind of framework, how it leads to this closed loop development cycle, which, as Messiah so nicely put it in his talk, allows neurofeedback and neuroscience to evolve together. For all these reasons, I'm very excited about fMRI neurofeedback as an avenue for future therapeutics development for mental illness. And my group has been very focused on it for a decade and a half. I currently have a very small team, but absolutely wonderful young people working with me. And we collaborate with many other people across Yale on different lines of research. So we have a line of research focused on OCD. I'm showing some of the people who've contributed very dramatically to this research. There's actually a humongous team. I can't represent everyone here, but these clinical trials take a lot of work and a lot of people to conduct. So I'm sorry I can't acknowledge everyone, but this is just a subsample of people who are working on this project. We also have a line of research focused on PTSD. Again, this is a small subsample of the people involved. We are currently running a clinical trial in adolescents with Tourette syndrome, hoping to improve their tic symptoms. And we're hoping to begin soon a study in borderline personality disorder. This will be in collaboration with Sarah Feinberg and Sue Whitfield-Gabrielli. So all of these lines of research have received funding from various sources. And a lot of these grants have allowed us to get new lines of research started. But the bread and butter, the large support for all of this line of research in my lab has come from NIMH. And I'm really grateful for all of the support NIMH has given for what I see as a very promising line for therapeutics development. I'm not going to talk about the results from these specific clinical trials we're running because they are all publicly available. We published all our papers and they are available on PubMed Central. So if you just go there and search for Hampson, the disorder you're interested in, neurofeedback, you will find our papers and the results. But to give you, my nutshell summary of it, I think that the results have been promising, but it's also clear that more work is needed to optimize these interventions and really get the best bang for our buck. And that I'm going to talk about more now in the second half of my talk. So I'm switching over to talk about challenges in fMRI neurofeedback early stage research. Actually, I'm just going to focus on one challenge because I think it's the major one of interest. And to understand this challenge, you have to think a little bit about what you think a healthy research pipeline should look like. So this is what I think a healthy research pipeline should look like. In the early stage, we should be completely focused on exploration. So these are studies where we're going to sample parameter space broadly. We want to find the most promising options. We don't want to worry about false positives because they're going to come out in the wash later on, but we want to look everywhere. The main thing is we don't want to miss something really promising in this early stage. And these are usually small sample size studies because nobody wants to throw, billions of dollars at something you've never tried before. So you expect a lot of small sample size studies in early stage exploration. Then moving on to middle stage is where you're going to separate the false positives from the real. So let me give an analogy of this. So say I thought there was some island in a good climate zone that it might have therapeutic plants that have a special compound. And you can take those plants and test them, investing lots of money and time in doing so to determine if they actually have the compound. But first I wanted to sort of find out if this island has promise for harvesting these plants. And I said, I want these people to go and explore the island, right? What we know about the plants is they live in swampy areas, they have spade-shaped leaves, they like a certain type of soil. Please, go to this island, explore, and see how much promise it has. Now, if you were one of these explorers and you landed on the island, what would you do? Is the first thing you would do go and pick the plants you see, right, getting off the boat and take them back and start doing time-consuming tests on them? Probably not. Probably the sensible thing to do would be to get the lay of the land and walk around the island and find out where are the swamps. And does the soil look like the right type of soil, if that's your expertise? And, did the plants have spade-shaped leaves? And so, all these different people are doing these little jaunts around the island, and they're sending back reports. Oh, it looks like there's a swamp in this area. And maybe that's a false positive. Maybe that swamp doesn't have any promising plants, but that's okay, because the second stage of research is going to winnow out the false positives from the real. So you're going to get a whole bunch of reports saying, oh, there's a swamp in the northeast corner of the island, and the northeast corner of the island has the right kind of soil. And look, all the plants have spade-shaped leaves. And then you start to say, okay, this northeast corner of the island seems really promising. So this is the middle stage of research, where you're looking for findings that crop up repeatedly. You're looking for convergence of reports across groups and contexts. Meta-analyses can be very helpful in this stage. But essentially, you're trying to figure out what is the most promising thing to take into the late stage clinical trials. And so maybe you focus on plants from the northeast corner of the island, you go there, you get your samples, and then you take those back. And that's when you do the expensive, time-consuming, well-controlled testing with rigorous statistics to avoid false positives, and using very large sample sizes to ensure you don't have false negatives. So that's classic late stage research. So what I'm getting at here is that the pipeline should involve a continuum, where the early stage of the continuum is exploration focused, and it's not focused on power, it's not focused on statistical rigor, it's focused on making sure you don't miss something exciting. And then, as you move along the pipeline, you get more interested in power and statistical rigor. And when you get to the late stage research, you're completely focused on power and statistical rigor. You're testing one thing and you are not exploring at all. That's late stage clinical research. What I see as the big problem right now, well, I'll get into that in a second. Let me just highlight a bit more this continuum, because I think it's an underappreciated point that power and exploration trade off against each other. For a fixed amount of resources, you can either test one thing really thoroughly with a very well-powered study, or you can look at a lot of different things, and you're not going to be well-powered for everything you're looking at, but you're just getting the lay of the land. And so, if you're going to be well-powered, you're not going to be able to do exploration, and vice versa. And so what I see as the big problem at NIH right now is that they've put these clinical trial mechanisms on as a blanket over all therapeutics development. And the clinical trial mechanisms, as they are structured, pressure the work to conform to what's healthy for late stage clinical research, but is extremely unhealthy for early stage exploration. So the clinical trial mechanisms don't really allow us to explore. Everything is considered a clinical trial that we do with neurofeedback that is intended for therapeutics development. And clinical trials are forced to be well powered. There's actually a power section required in the human subject section, as well as a statistics section. So when you put those sections as required parts of a grant, it tells reviewers that these are important criteria that should be considered in ranking the grant. And so you can't even submit a grant that's not well powered with any hope of getting it funded. And yet to do exploration, you have to accept that it's not going to be well powered, that you're going to use your resources in these small studies to explore. So basically power trades off against exploration. We're not really able to explore. And so much of our parameter space that, both Stephen and Messiah, as they talked about, we have a humongous parameter space to explore. So much of it is unexplored. And this is a huge shame. So you might say, well, isn't this a general problem? Like, why is this specific to neurofeedback? I think this is a general problem. I think that the way that the clinical trial mechanisms at NIH are structured is unhealthy for clinical research in general and clinical therapeutics development in general. But the other kinds of therapeutics development often have their own avenues for exploration. So for example, the pharmaceutical development relies very heavily on animal models. They can refine exactly what compound is best and address a lot of exploratory questions in animals, which obviously we don't do. And in the psychosocial therapeutics development world, they rely heavily on clinical wisdom, which is obtained by clinicians who spend years working with patients and try a lot of different things and get a sense of what works better. And that's a form of exploration. So they can kind of distill their clinical knowledge that they've gained from that process, which we also don't have that avenue to explore. So it hits us particularly hard in the neurofeedback research world. Whereas I think a healthy development pipeline would look something like this for us, where we're doing a lot of exploration in the early stage, and then in the late stage, we're just doing confirmatory testing. The reality is, and we're, because we're doing a lot of exploration in the early stage in an ideal pipeline, we would be finding the best interventions, and they would be identified in the middle stage and end up going into the tests, the confirmatory tests. But unfortunately, instead, what's really happening is because we can't explore, we land on the island and we have to just take one of the plants that's in our field of view. So we look around to see if any of them have spade-shaped leaves, and we grab them and we take them back and put them into late stage confirmatory testing. Well, it's not late stage, but confirmatory testing styled projects. And, this is a huge shame, because we're running confirmatory trials on suboptimal interventions. Better interventions are probably out there, but we haven't identified them. And this is going to look really bad for neurofeedback, because most likely a lot of the confirmatory trials are going to be disappointing because the best interventions were not entered into them. So in general, I see this as a huge shame. And I'm just going to throw out a few possible solutions that occur to me. One is that NIH could distinguish between early and late stage clinical trials in their funding mechanisms. Early stage clinical trial research should prioritize exploration and de-emphasize power considerations. So this would mean explicitly removing the power and stats from the human subject section. To be honest, the entire human subject section, this idea of a rigid protocol where you can't change anything as you go, is implied by having to define everything in advance in the human subject section. It's all designed for late stage research. It's beautiful for late stage research, and it's totally counterproductive for early stage research. So, you could just get rid of the whole human subject section, and that would actually be an improvement in my mind. Another thing you would really need to do is explicitly prioritize exploration over power in the wording of the mechanism, and ensure SROs communicate this to the panels, because at this stage, we have an ingrained mindset where everybody thinks that power is important for everything, and large sample sizes are important all the time. And it's just, I think, really counterproductive to early stage research. And we need to change the ingrained mindset now. Another obvious potential solution is to increase the size of R21s, which are the NIH's flagship mechanism for early stage research, and haven't increased in size for decades. So they're now worth, because of inflation, they're now worth a fraction of what they used to be worth. And they're just not large enough to run, to do much exploration, at least for fMRI neurofeedback studies. You can't do much exploration with the amount of money in an R21. And then the third suggestion is that we could consider mechanisms that provide greater flexibility, that fund investigators pursue lines of research rather than specific projects. And one of the concerns about going this route is that a lot of the senior investigators might get all the money, right? And that would be bad for development of younger investigators. And I think that definitely has to be actively avoided. A beautiful model to look at is NIGMS's MIRA R35. So they fund lines of research, but they make an explicit effort to make sure that they're funding people at all different career stages. So they're also protecting the career pipeline. So I thought that was really a nice grant mechanism when I read it. So that wraps up what I wanted to say. I just wanted to take a second to talk about some things that may interest viewers. So first of all, for those of us who do fMRI neurofeedback, the big meeting is called Real-Time Functional Imaging and Neurofeedback, or RTFIN, and it's going to be held next in Heidelberg, Germany in early November. But we would love to see you there, RTFIN 2024. Also, if you're new to fMRI neurofeedback, you're a researcher thinking of moving into this field, we have recently published a textbook, which is designed just for that purpose. This was an international group effort with a lot of the world's experts on fMRI neurofeedback contributing, and it's really designed to be didactic and to help people fill in gaps, because people come from all different interdisciplinary backgrounds into this field, and so everybody's got gaps somewhere. So if you think you might be interested, check it out. Thank you.
Thank you, Michelle, and thank you, Stephen and Messiah. We have a lot to cover, and you guys have given us a very nice kind of overview of early stage research and some of the complexity that's involved, and also the challenges and the types of research, securing funding for that research, but also just the methodological hurdles to overcome. We've gotten a number of questions in the chat, and thank you to the audience members and viewers for submitting those. I'm going to try to hit as many of these as possible, but also I want to open up the discussion to our panelists who may also have questions and want to riff on this a little bit and provide their input. The other thing I just want to give folks a heads up is that we anticipate a lot of overlap between these sessions, because I think a number of the researchers that you see here today are conducting research both at early, mid, and later stage investigations. And so I think there's going to be a lot of input there. But one of the first questions that I wanted to ask, and this is specifically for Stephen, but I suspect other people may want to comment on this as well, is that we've received a number of questions in the chat about control conditions and the selection of control, the pros and cons of choosing controls and the types of controls you choose. And Stephen, I wondered if you could go back and expand a little bit on that and the issues that you see, particularly from an fMRI researcher, in how they relate to the choice.
Okay, yeah, thank you very much, Chris. And, first, I think I should say what Michelle was saying captured virtually everything I wanted to hit in terms of positive controls, in terms of early stage exploration and sampling the parameter space very broadly. Once you start to sample that space, what I wanted to say was then you can branch out from there and kind of have a safe zone. When you start, in this case, I'll use a slightly different metaphor. When you plant the seed and it doesn't grow, there's nothing to control for. You don't have to control for the rain or the soil or the sunlight, because you first need to sort of find these promising results. And so I could share my screen again and just kind of go into that Control Freaks paper, which has this, I just happened to pull it up because I suspected there might be a question because there seemed like a lot in the chat on this. So I think I'm sharing. Can you guys see my screen?
Yep.
Okay. So, what's shown here is a nice flow chart, and that dovetails, I'm using dovetail a lot, but that meshes perfectly with this table. And then, of course, if you read the paper, it goes into a lot of detail. I think at the end of the day, the message of this paper, which was not very controlling or super didactic, I found it to actually be very thought-provoking and sort of open to flexibly interpreting what you need. At the end of the day, what I would like to say about controls is once you start to see results and start to get into the later stages, and Michelle and Kim may want to weigh in on this, then you want to control for as many things as you can think of and afford that may be sort of negating the fact that the real-time fMRI neurofeedback itself is sort of causing these very positive effects that you're seeing. But as reviewers and as investigators, we also have to take a step back and take responsibility for figuring out what those controls should be. And so these flowcharts are very helpful if you use them as kind of an exercise that you generalize on your own. If you start to really cement them or concretize them without taking a step back and figuring out the context of the experiment and what its claims are, then you start to get into this formulaic view that I personally feel is a little bit dangerous and kind of reminiscent of using p-values of 0.05, which is kind of an arbitrary thing, and then people just take a break from thinking and just say it didn't surpass 0.05 statistical significance, so it's not significant, without going the step back and thinking about what positive and negative, what false positives mean and what power means in those contexts. So that's my take on controls. I think you as an investigator should be responsible about thinking about what else could be causing the control. Once you learn something from an experiment, that doesn't mean that just because that real-time feedback was not important, you may actually learn how to do some synergistic things outside of the scanner through your real-time control experiment. So I would just say, try to take on some responsibility. That's for me as well as everyone else. We need to think about the context. Michelle, do you have any?
Yeah, I mean, I'll echo this idea that this obsession with P0.05 and statistical rigor is really not terribly appropriate for early stage research, where just the sheer information you're getting, the lay of the land, is what we really need to get in the early stages. I agree.
Yeah, and I also think that unspecific effect is there are so many unspecific effects in the real-time neurofeedback. So we cannot address only the one control condition by every unspecific effect. So it's a kind of control condition is a kind of a null hypothesis. So rejecting that control condition may prove the active conditions, but we also need to find the active condition is really working or not. So it's a kind of mechanistic understanding of the active condition is also important to validate the current interventions or neurofeedback.
I will say that time helps with some of Michelle's exploration. It's not going to fix the sort of NIH mechanism approach or the clinical trials structure. But as we grow a community, I think that's also an important effort and part of the benefit of meetings like today. And thanks again to NIMH for hosting this.
Thanks, Stephen. Let me ask just a follow-up question, because it was this is a question that we received in the chat, but it relates to this idea of exploratory research. And the question is this: exploration is lovely if we can detect results when we have them. I like the recommendation to allow more mechanisms which provide for exploring in larger samples. I'd like to know more about how doing underpowered research, even in an exploratory context, allows us to detect interesting potential results, if that is recommended. I see how, if we want to restrict ourselves to detecting large effects, we can power for detecting those with smaller samples. Or how can we do repeated measures testing of multiple targets with larger samples and allow for having different families of tests? But I'm not yet seeing how abandoning power lets me make scientific conclusions.
One thing comes to mind immediately is we need to push to share data. As we share actually raw data, we can. And, I also think that these are challenges within the publication realm as well. How do we publish data that are underpowered and convince reviewers, or even convince ourselves that we have data that are worth publishing? But as we share data, we can go back and scrutinize those data, compile those data, amass them across different centers to do it. So that's one thing that comes to mind. Obviously, the question is quite a challenging one, and we may not have exact answers for all of it. Michelle, do you?
Well, I think that the question is undervaluing the importance of replication in the early stage. So I think that, yes, you're going to get false positives. You don't conclude anything from a single false positive in an early stage research study, but you look to see what emerges as showing up again and again in the literature. To me, that's how you find what's real and separate it from what's not real in a healthy early stage pipeline. So I think there's this misguided notion now that we have to know if it's real based on a single study, and that you can't publish anything unless you know that that's real with great confidence. And I think that's a problematic mindset, that we need to say, no, you can put out, there are going to be false positives, and that's a healthy part of the pipeline that you accept, but you have to also read the literature with recognition that these are small sample studies and they have to replicate a lot before I'm going to invest a lot of money in going forward with that as a late stage trial.
Yeah, I believe the false positive is a part of science. So of course, we need to validate later more complementary analysis, but we can start with some findings. So if it is either the false positive or real positive, so yeah, we should start with that.
I mean, what do we think about false negatives? That's also a challenge. And then
And I think that's worse in the early stage.
Yes, much.
Another question that we received what are your thoughts on screening potential participants on their ability to up and or down regulate brain activity prior to enrolling them in a study, considering that several studies show wide variability in subjects' ability to regulate neural response? Fetty et al. In 2020 reported that attention, motivation, and mood can be important influencers of neurofeedback performance. Should these factors be assessed in all neurofeedback studies, even if they aren't necessarily the specific phenomena of interest?
Well, one thing that comes to mind is in the past when we've done multi-session neurofeedback studies, on the initial assessment day, this was work Pearl Chu and I were doing. On the initial assessment day, we actually put people in the scanner for half an hour and had them do a really simple motor neurofeedback task. And essentially we had staged enrollment in the study. If they were not staying still in the scanner and couldn't control kind of an interface using really simple motor tasks, then we didn't pass them on to the next stage of the study. So that was used as kind of a filter, but also kind of an exercise in training participants about kind of the time sluggishness of neurofeedback, given the hemodynamic delays that are inherent in this neurovascular coupling. So that's one approach, would be to sort of screen participants that based on their ability to be still in the scanner and teach them what neurofeedback, what they can expect in a neurofeedback experiment.
Thanks. Another question. A large issue in fMRI is related to the knobs is the choice of software. Could you talk a little bit more about software packages that you use and whether any comparison has been made between these to address reproducibility? This may be just as much of a problem as the choice of control condition.
Well, I do think, again, in terms of reproducibility, offline analysis is useful. So, you know, my group started off directly programming the Siemens scanner, and then we realized that that was impractical and we were going to have to reinvent the wheel. So we built a plugin for AFNI, which already had real-time capabilities. And so our plugin in AFNI is running during the experiment, but once you are done with the experiment, those data can be reanalyzed in SPM or FSL or any other package that you want. Now, there are a lot of software options, which I think is good, and it ties back to growing the community as well. Like, we need more software that's available. One cool thing that AFNI has is the ability to replay data through your real-time pipeline. And so if you have a nifty file, you can essentially feed it through a command called RTFeedMe and simulate the scanner collecting those data. And so that's one part of an answer to that question.
Yeah, I also implemented our own systems for the real-time neurofeedback, but it's also based on the AFNI systems. And I think that it's so between the availability or accessibility and the flexibility is a trade-off. So some software is accessible, so very easy to use, but it's a small room to optimize or customize for each individual experiment. So there is no standard way for the neurofeedback yet, or no standard software. So yeah, there are some commercial products, yeah, but I think that many labs using their own in-house implementations.
Another question that we received. So you talked about the sharing of data as being important, and just for simple, where do researchers in neurofeedback share their data? Is it in Open Neuro? What other repositories might exist or can be encouraged?
Yeah, I think Open Neuro is a great option. With Cameron Craddock, we've shared data through kind of the Rockland study site as well. So I think if you share the data, they will be findable.
I'll chime in about the whole data sharing thing, though, because
yeah,
I feel like there is a big emphasis on data sharing, and there's a lot of people now whose careers are pretty much focused on analyzing data that's publicly shared, and they're not doing data collection. and it's much easier for them to, they can just download this data, analyze it, publish. It makes it very hard to encourage young people to learn to do the data collection and to learn to design well-designed data collection experiments. It's almost a sacrifice in their careers because they can't compete, of course, in terms of the publication rate with just downloading and analyzing publicly available data sets. So I think there also has to be an effort to try to get credit to the people who've collected really unique data, difficult to collect data. They have to be included somehow as authors. It has to further their career. Otherwise, we're not going to be supporting that line of research and the ability to do this kind of unique data collection.
Another question that I have, I'm actually going to combine two questions that just simply use the same word, and that is replication. And the two-parter of that is, first of all, how do you decide on how many replications are needed before the field has established that a signal is real, positive, reliable, and significant? And then flipping that, do you have any suggestions or comments on how many numbers of sessions of neurofeedback training are sufficient, as fMRI scans are quite expensive? So there's the reliability of a signal, replication of the signal, and then there's the replication and reliability of treatment. And I know I'm cheating by putting this together, but could you comment on those aspects?
Anecdotally, it says real-time fMRI neurofeedback needs a fewer number of the sessions compared to the EEG neurofeedback, but it depends, of course, on some target function or target populations. So, for example, in our studies, targeting the MDD participants shows a very quick response to. So Kim may have some comment about that there. But if we apply the same protocol to the PTSD participant, it takes more sessions to control the left amygdala activities. So it may depend on the population and approach.
And I'll just throw in as a kind of a preview, I will be addressing how we establish the number of doses that we give for our real-time neurofeedback intervention at my talk in the second session, and some of the issues that neurofeedback researchers can consider more broadly when determining dose.
Thanks, Kim. Another question, actually, you talked about sharing data. Are you also able and willing to share AI algorithms and model cards for researchers?
Well, for our AFNI plugin, that's shared. So we have access to AFNI's Git repository, and we do a medium job of keeping it updated. But certainly the crux of the capabilities are shared. And then there are some specific communication parts that are specific to what each site has available, and also if it's a GE, Philips, or Siemens site. So it tends to be a little bit difficult to get sites up and running, even with the free software to do so. But we do it on kind of a site-by-site basis to provide support.
Yeah, I have to say, I think the fMRI neurofeedback community is really great about sharing software. It seems like everybody's willing to share their software, and they're very helpful, if you ask questions, and there's a lot that's publicly available just on the web that you can take.
Yeah, I also shared my software on GitHub, but my battery, so documentation is not so great. So yeah, so most of the time, so
That's usually the challenge.
Another question, thank you. And this, by the way, other folks I think might want to comment as well. And that is, given the evidence that multiple neural network strategies might result in improvements in behavior and symptoms, how might we leverage a technology like TDCS in conjunction with real-time fMRI, given that TDCS modulates broad brain networks in a way that other neuromodulatory methods do not?
Yeah. So yeah, we can combine the real-time monitoring of the brain activation with the online stimulation, so optimize the parameters. So yeah, so I think even not only the TDCS, but focal stimulation like a TMS also affects the whole brain activation patterns. So I think we need to see the whole brain activation patterns or other regions effect even in the focal interventions. So yeah, there is a possibility to combine such a stimulation, external stimulation and real-time neuroimaging to optimize such parameters, I think.
We were really interested in the idea of using TDCS to try to amplify neurofeedback to see if you could, make those areas, learn quicker because of the TDCS. So I think there are a bunch of kind of combined applications that could be really interesting.
I agree, and I think the whole idea of combining a lot of these neuromodulatory interventions to prime the neural system that you're subsequently going to be intervening on, or to sustain the clinical benefits. For instance, real-time fMRI neurofeedback could be used synergistically with, say, ketamine, for instance, where we know it doesn't have a sustained clinical benefit necessarily. Or you can use it, to your question, TDCS to prime the neural circuitry for real-time fMRI to increase treatment efficacy. So I think that's a fabulous question, and I think a lot of people who do neuromodulatory interventions might be siloed, but I think we need to combine them either simultaneously, which we'll talk a lot about, and Michelle and Kim will be talking about too, or synergistically in a serial fashion to prime and sustain benefits.
You're muted, Chris.
Thank you. Following up on what Michelle said about pharma research having animal models as a starting point, do you think animal studies could contribute to neurofeedback research, for example, guiding exploratory trials or validating imaging studies?
I dare to say yes. I'm just going to jump in. I know that there are already technologies that can actually somewhat intrusively monitor animal behavior in real time. I mean, there are micro electrodes that you can basically insert in the brain of the rat, and you can actually, specifically provide a kind of stimuli that will give us in-depth mechanistic circuitry pathways and very much time by time exposure to a stimuli to circuitry. And some of that research has even been done, and I know some of the people that do that at the University of Minnesota, if they're still doing that, but I know they were a while ago. So those are my 2 cents.
I think on the physiologic side, to understand what's possible is good, but I do think animal studies are challenging. First of all, you need to have an animal model of whatever population that you are interested in, and then how do you coach them in terms of cognitive strategies, or how homogeneous or how conserved are the networks and even regions that are involved for that. So and even just the going back to Michelle talking about the idea of it's being natural learning, neurofeedback being natural learning, what are the reinforcers then for an animal model versus a human model of the disease? And so, the brain as behavior, I think, is a powerful thing that gets unlocked with real-time fMRI. But the difference then going from human to animal models is the gap is huge, I think.
Yeah, I think there are some interesting things that you could do with animals that would be relevant for fMRI neurofeedback, but I don't think the bread and butter early stage exploration can be done in animals.
Yeah, but physiologic limits, I think, would be useful and a good example.
Yeah.
And also the difference between the animal and humans. In human neurofeedback, usually the modulation is done with voluntary process, but we cannot do in animals. So in animal model, we reinforce some rewards. So it's a kind of the implicit learning. So it may be different from the human.
I have a question for Michelle and Messiah about mechanisms. What are the mechanisms for acute versus long-acting effects of real-time fMRI neurofeedback? Do we have longitudinal studies that can tell us this?
Yes, for me, I don't have so much data about the longitudinal data set. We have only a week or a few weeks after the training. So as Michelle presented, so interestingly, the treatment effect is enhanced as the time going. So yeah, so Michelle have some comments about.
Yeah, I would say that we don't have an answer now, but it's a super interesting question that a lot of us are interested in, and we're now trying to collect data. At first, we weren't collecting data, at follow-up brain imaging data, but now we're often scheduling brain imaging scans at follow-up as well to try to get some information about, what has changed in the brain since the neurofeedback ended. So we get it right after neurofeedback, and then we get it maybe a month later. So it's a very interesting question that we'd all like the answer to.
Great. Questions are coming fast and furious. Thanks everybody. And this is one that we likely other panelists will want to inform. We'll be talking about later, but we often see practice effects or challenges with near versus far transfer and cognitive training. How do folks think about overfitting in the training context? Are there unique challenges and opportunities related to near and far transfer effects in fMRI-based neurofeedback?
Can I just clarify what the language near and far transfer effects? Does this mean transferring to something that's very, very, very similar versus transferring to a task that's somewhat different?
Yes. By the way, and since this question came from one of our other members, Luke Stokel at the National Institute of Aging. Luke, you're welcome to unmute and ask a follow-up if that's helpful.
Yeah, no, you're right, Michelle. That was it. Trained to the same thing. I was also thinking about the outcomes themselves. Often, we're using similar outcomes where there might be practice effects. So I wonder how those things are accounted for in the context of real-time fMRI, and if there's anything unique about real-time fMRI neurofeedback and related to those issues.
I mean, I think that these are like all things that require more work for us to map out. I was really surprised by some of the basic science studies, which suggest very targeted effects, which in some ways is really impressive, that we can have these very precise, narrow targeted effects and not affect other things, so that we're not like drugs affecting the whole brain. But on the other hand, sometimes we want things to kind of have a little broader of an impact, so they affect you in a more general circumstances. So I think there's a sweet spot, and it's really an empirical question, how well we can tailor it to that. But I think one of the features of neurofeedback is this wonderful development, loop cycle where if something's too narrow, we can try to think of ways to broaden it and look at the neuroscience for that. So there's this chronic ongoing optimization that we can engage.
Thanks, Michelle. One follow-up question. So earlier on, Michelle, you talked about the challenges. And I think all three of you guys talked about the challenges of early stage research and the challenges that come with power and also finding suitable mechanisms that kind of match up to that. So at NIMH, we have a U01, which is a cooperative agreement mechanism for early stage device development. And I'm wondering if those kind of cooperative agreements or early stage device NOCs might be applicable to the kind of work you're doing. And if not, then I think you're also highlighting some of the other challenges too that go on, and maybe if you could expand on that a little bit.
It's hard for me to comment on those because I'm not super familiar with those mechanisms. Mm-hmm. And are you thinking neurofeedback would be considered a device in that case, or that the mechanisms would be broadened to include? Because currently we're classified as psychosocial.
Yeah, that was going to be my point, is that we currently are not considered device-based intervention and not considered part of the device-based community. At least we have not been up until this point. And I will discuss that a little bit in my talk as well.
I'll just hop in here and welcome you to the device-based community. As we'll be hearing in session 3, neurofeedback is a device, whether it uses fMRI or EEG. And actually, that is part of the purpose of this discussion. And the U01 mechanism, just for folks to know, does specifically address the early stage needs for both drug and device development. And the emphasis is on exploration. The emphasis is on discovery. Some of the work is first in humans, so it is very, very early stage. And we will be happy to put a link to that in the transcript from the meeting.
Is it a large grant?
Yes, it's not limited in size. I will also point out some of our mechanisms, when you need to exceed $500,000 per year, permission can be requested in advance for that. I'll also point out, you were mentioning the R21 as a high risk, high reward or early stage work. And for studies that are seeking to develop an intervention, those actually need to go to the R61, R33, at least at NIMH, which does not have a cap on its funding, whereas the R21 does have a funding cap.
So one of the things I struggle with, you use the term first in human, because most of the exploration has already been done when you get those mechanisms. So that's the problem for us with those kind of mechanisms. We're competing against people who've done tons of exploration and have a lot of data supporting their intervention, and we haven't been able to explore. So that's difficult for us. We need mechanisms that actually just support early stage exploration. They need to be big enough that we can actually do some exploration, but we can't be competing for these really large grants where you're expected to have a lot of data already supporting you. That's kind of the challenge for us, I think.
Yeah, so I will point out, I will encourage you all to look at the UO1 mechanism. It is meant for early stage. And when you're dealing with a device that is not FDA cleared for the treatment of a psychiatric condition, which currently real-time fMRI neurofeedback is not, that is considered early stage because it's not currently clinically approved. And much of that work is to figure out how to dose the tool. I think that we learned today that there are a lot of knobs, there are a lot of parameters. And the early stage mechanisms are meant to help investigators work out those dose response functions to figure out are you going after a region of interest or are you going after a network? How do you develop and optimize the machine learning or AI approaches to extract and decode the signal? How to do the timing of the feedback. We learned from Stephen Lacan's talk that timing the feedback is very important. How to select the controls. Those are all considered early stage questions, which we do want you to work out before then using the tool to interrogate a target. And the R61, R33 is really about target engagement and interrogating that target. We want to know before you get to that stage that you have good evidence that the tool is likely to be able to engage that target. And that's where the early stage work is meant to be done before you go to our R61, R33 phase.
Thanks, Holly. Thanks, everyone. We're actually ahead of time, but I wanted to just and so maybe we can have a little bit of a longer break. But I did want to ask a more general question, and we've received some questions from folks who are asking kind of this general question, which I think a number of you have already answered. But I'd like to return to that, and that's a question of why fMRI based approaches now, and if you could expand a little bit more on the benefits or pros of fMRI approaches, particularly as they may not only inform treatment development, but also they might inform this larger question of helping us to understand the mechanisms of disorder and how this tool contributes. And so I guess that's another question. I mean, there have been other approaches along the way, but why fMRI now? Why is this exciting?
Well, I think fMRI allows you to translate from, basic neuroscience. It's very easy to figure out how basic neuroscience translates to fMRI. And so you can be informed by a huge, rich body of information. And then it also, as people have discussed in the session, it gives this incredibly rich mechanistic data as you're running it. So it really allows you to do continued refinement of your intervention and targeting it better and better in a closed loop kind of development cycle. More so than other imaging modalities typically do. So that's my take.
Yeah, neuroimaging study with fMRI is increasing, and the neurofeedback interest for the fMRI neurofeedback is increasing with its development. So I think that, so as I said, so neurofeedback evolved with neuroimaging evolved, I think. Yeah. So that's why fMRI is now interested in that.
You're muted, Chris.
Versus technology. Thanks. Michelle, you had mentioned that there's a value in real-time fMRI neurofeedback for youth. Can you tell us how much work has been done in children and adolescents? And I also suspect others may be commenting on this as well.
There's been a fair amount of work. Karina, I'm sure, is going to talk about some of her work in the next session. And there's been some work in the UK on kids with ADHD. We're doing adolescents with Tourette syndrome. course, most of the work that's going on is older kids because it's very hard to get little kids to stay still on the scanner. But those are the things I'm most aware of. I don't know if others can chime in and mention other things.
Yeah, I just wanted to reiterate what Michelle has stated, that the initial steps were primarily done in ADHD in King's College, London. And there seems to be a healthy balance of both positive and not so positive results. And that's just a way of generating valuable data. But I believe that the field in children and adolescents is very much expanding to other conditions. And that I'm very excited and honored to present here later.
I'll add also to what Karina said, and that in collaboration with Randy Auerbach's wonderful lab in Columbia, we're trying to target repetitive negative thinking like rumination in teens with depression. So I think the field is growing quickly and we're quite excited about it.
Thanks. And it's a little preview of what's to come. I did have a question that was submitted for all. So Messiah argued persuasively that understanding mechanism is key to figuring out how to kind of set the knobs that Stephen told us about. What do you see as the highest priority areas for research to address unanswered questions about how real-time fMRI neurofeedback works?
Mhm. Yeah. So at least for my personal interest is a mechanistic understanding or so more neuroscientific interest for the neurofeedback effect. But of course, it's important as an intervention method, so that we should focus on the outcome measure, the cognitive functions or symptomatic or therapeutic effects. So yeah, so I'm not sure so it's a universal interest, but at least for me, it's mechanistic understanding is my current priority.
I agree, and maybe I'm slightly adjacent to what Misaya is saying, but I'm really excited about real-time fMRI's capability to further refine our understanding of functional roles of regions and networks in the brain. And so what you end up with is an experiment that can fail. And so you have a stronger falsifiability through this closed loop experimentation than you do in a sort of open loop fMRI setting. And so what that means is that you can start to scrutinize aspects of your task or your pipeline and understand how that affects ability to neuromodulate. So if you give, if the literature points to 10 or 12 cognitive domains for anterior cingulate, you can start to take tasks or task strategies from those different domains and do head-to-head testing to see how that maps onto neuromodulation. And then you can study your data post hoc as well and see, network or broader downstream changes that happen in that case. So that's.
Yes, Tama, Dr. Hendler.
I would like to address here something that I think is important to remember, that neurofeedback is actually establishing an association between a brain state and a mental state. And as far as it's important to study the neural mechanism, that there are two levels of learning that are occurring simultaneously, actually. The brain learns, so to say, and the mental state is also being modified. And I think these are two paths that are very important to study and to put effort in order to improve the efficacy and the outcomes of neurofeedback. And we should remember that we have these two levels. And I think for the neural level, I would like to contradict what was said. Actually, animal work might be very, very valuable, but also repeated sessions in humans might allow us to learn a little bit more about the reinforcement learning or other learning mechanism that takes place. But at the same time, the mental operation that is being formed while we are trying to affect the brain is also something that is being learned. And that's very open yet to our understanding.
Thanks, Dr. Hendler. We have time for a couple more questions. I do ask for one, and then we'll go back to the chat and try to grab some too. We talked about a little bit about software and algorithms. I guess this maybe for Messiah and Stephen, what work is needed to be done in the software space? What are the key questions to be addressed about signal extraction to train the modulation of distributed networks?
Yeah, fMRI signal itself is a very slow, so has some several 5-second delays. So I think at least for it should be shorter than the 1 PR or 1 volume acquisition. So after that, the faster is not so much advantage for the fMRI, at least for the fMRI signal, I think.
Well, and something Messiah was saying earlier, I'd like to kind of expand on, which is sort of the documentation and the complexity of the software. And so, getting this to be more plug and play would help us to expand the community. We didn't get into the nuts and bolts of setting up a real-time fMRI system, but at the very least, you need computers that are communicating with each other and having access to the data very quickly after acquisition, either reconstructed or unreconstructed form. And so right now it's best to have a dedicated system or machine that's there, but a lot of groups, because of policies with the scanner, may have to resort to bring in a laptop in, and then you have to test all these things. It just adds a much more experimental complexity and kind of a sense of, well, this experiment might fail, or it's way too hard to get done 6 months later, after we set it all up. So I think on the software side, getting things a little bit more plug and play would help a lot with everybody being able to do these, making this accessible.
Yeah, so we need to work with the MRI vendors, so Siemens, GE, so to implement the real-time data explosion. So that part is, I think, the bottleneck of the most real-time systems. So data transfer from the scanner computer to the external processing computer is actually the most time-consuming part now. So I hope the MRI vendors will help us to implement such a standard protocol or some more time-efficient real-time implementation of the data export.
Yeah. We have a very tight coupling with the Siemens system and the AFNI system. So we actually make it so that the operator of the scanner can just run it, and as much as possible is automated. But that requires a lot of scripting. And so then those scripts become hard for other groups to read and maintain. Even if we're sharing them, that's a lot of work. And so they can take what we develop and we can help them to try to generalize it. But it is a challenge. So the software side of things is a challenge.
We're actually at time for a break, and so I apologize I can't get to all the questions, but I did want to ask just one more question, or at least, and that is, a number of folks have asked about the role and the long history of EEG related neurofeedback. And one of the decisions we made in putting this meeting together was to specifically think about and talk about fMRI informed real-time fMRI neurofeedback approaches specifically, because it's where we're seeing a lot of new research and a lot of new applications. And I'm wondering if you could comment a little bit on the history of EEG research, and I also and how you think about this modality as it relates to other modalities like that. And I would also, just for those people who are asking, I just want to give you a little preview too for session 3, when Dr. Thelma Hendler is going to talk about her work, which actually thinks about the use of fMRI, how it informs EEG delivered neurofeedback. But so general question about EEG and how it relates to your work in real-time fMRI neurofeedback.
Well, I'll just say there's a ton of interest in the field of, doing what Thelma Hendler has demonstrated, which is taking fMRI neurofeedback and eventually being able to translate it to other modalities and use it effectively with cheaper, more accessible modalities for the purposes of clinical dissemination. It's a kind of a gold ring in the field.
Yeah, I think that it's a way to translate the fMRI neurofeedback knowledge. So translate the EEG implementation is one way to more accessible neurofeedback approach. So I think the EEG neurofeedback has a long history, but in most cases, used signal is not so kind of limited compared to the fMRI neurofeedback. So fMRI neurofeedback can target a specific brain region in various parts, but EEG usually uses some so like a frequency measure or alpha band balance or something like that. So the variety will be limited compared to the fMRI signals. So if we can use such a variety of fMRI neurofeedback signal translated into the EEG neurofeedback, it could be more accessible and more effective treatment may be achieved.
Thank you. Thank you all very much. This has been a great discussion, and I really appreciate your presentations. We're at a break. Is that right, Producer Deborah?
Yes, until noon.
Let's aim for noon. So we'll take an 11-minute break, and we'll start session 2 at noon. So thank you all very much.
All right, so I can jump in while Chris gets his audio together. So I'm Alex Tolkowski. I'm also a program officer here in the Division of Translational Research, and it's my pleasure to moderate this second session here today. We'll be hearing from 3 presenters before we have our discussion. They'll be in order: Susan Whitfield-Gabrielli, Karina Cavedo, and Kimberly Young. I'll ask them each to pass the baton to the next presenter when they finish up with their slides before we'll go into our discussion after Dr. Young's presentation. So Susan, whenever you're ready, you can please go ahead and share your screen and take it away.
Thank you, Alex. First, I wanted to give a tremendous thanks to NIMH for hosting this phenomenal workshop. I think it's a really terrific opportunity to share ideas about what I think is a really transformative intervention, and that is real-time fMRI neurofeedback. Today, I'll talk about the challenges and opportunities of personalized network-based neurofeedback and neuromodulation in psychiatric disorders. So I think it's fair to say that we're facing a tragic youth mental health crisis, and not that long ago, the U.S. Surgeon General and leading pediatric health organizations have all declared a national state of emergency in adolescent mental health. And it's clear that we need urgent, innovative, personalized treatments that target the core mechanisms underlying mental illness. When I take a step back and I think about critical barriers that are preventing us from moving the needle in mental health treatments, I'm thinking of 4 such barriers. One is that the mental health care system can be primarily still oriented towards remediation instead of prevention. Treatments are often given on trial and errors, and we know that there's great heterogeneity in treatment response. Often gold standard treatments are not very effective. On 30 to 50% of patients often respond to pharmacological and behavioral interventions, and they often have negative side effects and high attrition. And when they do work, we don't necessarily know why. So the pathophysiology of mental illnesses are unknown. Some drugs were accidentally discovered, so on and so forth. But I think that real-time fMRI neurofeedback can help address some of these critical barriers. And today I'll be talking a little bit about how we can potentially use it for neuroprevention, neuroprediction, neuromodulation, and in the case of real-time neurotriggering. So we and others have showed that baseline human brain neuroimaging can predict progression of symptoms such as anxiety and depression years later. Even in a normative pediatric sample in children ages 7 can predict worsening of, say, anxiety and depression and internalization 4 years later. And it would be great if we knew in advance who might be more high risk, then we could actually offer things like real-time neurofeedback and help mitigate symptom progression and possibly even over illness. We and others have also shown that baseline neuroimaging can predict treatment response, but the advantage of real-time neurofeedback is that we can also look at neural flexibility and neuromalleability and look at the change of resting state networks pre-post the intervention that might be an even better prediction of long-term treatment efficacy. And of course, in the real-time neurofeedback framework, we're hoping that this real-time feedback, people will learn how to modulate their networks and have a subsequent modulation and mitigation of clinical symptoms and increase in cognitive performance. However, going through all of this process, it's really important, as we've heard about in the first session, to try to understand more and more about causal mechanisms. And we can do this in many different ways. We can do this by multimodal neuroimaging, say with MRS and fMRI. We can say look at how GABA and glutamate may be changing pre-post some of these real-time neurofeedback interventions. We can also use real-time in a different context, and that is rather than using real-time fMRI for feeding back activation or connectivity, we can use real-time fMRI to detect brain states, at which point we can trigger either fMRI task stimuli or experience sampling. And in that way, we can actually build predictive models so that we can identify brain behavior and brain symptomatology in a causal way. So understanding causal mechanisms, of course, will help refine these interventions and ultimately tailor our treatments. So how does this all work? Of course, we're all using an fMRI scanner, and she can look at brain structure and brain function. Today, I'll be talking about more the intrinsic functional architecture of the human brain as revealed by resting state functional connectivity. And here we're looking at ultra-slow frequency fluctuations that cohere into functional networks. And we call them resting state networks because they cohere in the absence of any task whatsoever. So in the red, you'll see regions of the brain, even if they're anatomically far apart, that rise and fall in temporal synchrony. Now, individual differences of these resting state networks, and importantly, intra-individual differences in resting state networks actually give great insight into brain health and disease. Luckily, these brain networks are plastic. It can be modulated with interventions like real-time fMRI neurofeedback. And what I hope to excite you about today is that real-time neurofeedback can actually use these entire network and network interactions in the context of neuromodulation. So we can build what we're calling network therapeutics. And so we believe that being able to modulate large-scale networks and their interactions with other networks will really provide a more effective method of neuroregulation than neuromodulation involving a single region or anatomically unspecific pharmacological interventions. But I'm going to take a pause here and just mention that this is really a tour de force. And, you know, this real-time neurofeedback offers many, many opportunities, but as you know, it comes with many challenges, many of which you've heard about already. And I can say, I think with some confidence that there are many, many challenges that we've run into over the last 20 years. We've been doing this since the early 2000s, and we're still really in the exploratory phase in many circumstances. We're still learning, we're developing new approaches, new algorithms, we're tweaking, we're optimizing acquisition parameters, analysis, and feedback methods. But the idea here is that when you're funding the real-time fMRI neurofeedback community, you're also not only funding for that particular project, you're also funding people who are developing a lot of really novel neuroimaging tools that can be used in basic science modalities, and I think address gaps and barriers in the field, open up new windows to understanding brain network organization and function and health and disease, and really paves the way for us to innovate biologically informed approaches, including real-time fMRI neurofeedback, but not limited to that. So
Susan, I'm
yeah.
sorry to interrupt, but for the sake of our closed captioning, could you please just slow down a little bit?
Oh, yes, it's a biological problem. I'm going to get real-time feedback to try to slow down, but I'm working on it. So in our case, we have something called vSend, which is a patch developed by Paul Whiteman and Andre Vanderko, MGH Martinos, and they have been developing this for many years, and they're maintaining it for us. But it takes a lot of work. In fact, right now, they've been working on it for quite some time to work interoperably with the Siemens upgrade, which many of you may have had some experience with, non-trivial. And so once you get the data, which you have to do very quickly, then you need to clean the data because, as you know, artifacts like motion artifacts can have a huge deleterious effect on the activation and connectivity that we're trying to look at. In particular, like motion artifacts can substantially affect things like the default mode network. Once we clean the data, then of course we have to analyze the data in a way that we can feed it back quickly, like incremental GLMs. We may look at activation, we may look at connectivity, but as you know from the first session, there's many different knobs to turn, and you do this and put it all together in this feedback scenario. And ours is open source called Murphy, initially built and by Oliver Hines back at MIT. And we're continually modifying this and upgrading it. And we using a system to do neurofeedback, but also neurotriggering. And I'll tell you a little bit more about that. But then eventually, of course, we want to scale this intervention. And so that requires simultaneous EEG and fMRI. We're trying to build something called a mind balance training platform. But again, that's a huge amount of technical development that's going on behind the scenes. So because it takes so much effort, it really takes a village. So in our case, it started in the early 2000s when Christopher DeCharms came to Stanford and asked, can people learn to regulate brain activation in specific regions using real-time fMRI neurofeedback? We didn't know, and so we tried it. And after a long time of tinkering, we were able to train people to how to modulate their motor cortex. And then he published his first paper in 2004. Then a more perhaps interesting question after you're convinced of that is, does learned regulation of activation or connectivity of these regions influence the mental processes mediated by those regions? And I think that's what a lot of us are working on now. When I moved to MIT, I had the good luck in running into Eden Evans and Luke Stokel, who did a lot of work with real-time neurofeedback and addiction. And they organized a beautiful conference out of which came this nice optimization, real-time fMRI neurofeedback for therapeutic discovery and development paper. Many of us are using real-time fMRI neurofeedback to help clinical conditions. And in fact, with Margaret, we were trying to help people with schizophrenia to reduce auditory hallucinations. We got an R21 to do this, but as Michelle was saying, an R21 doesn't necessarily cover the costs that are needed to, turn all the knobs, so to speak. And so we were working on this for quite some time, but it turned out that we actually needed to combine real-time neurofeedback with mindfulness meditation. And that was really the sweet sauce for us. And currently we're using that the combined modalities of mindfulness meditation and real-time neurofeedback in the case of depression with our colleagues from Randy Auerbach's lab. But why is it that we need this mindfulness meditation? Well, it all comes down to the default mode network. So this is an iconic figure from Fox 2005. We know the two core medial hubs of the default mode network are related to self-referential processing and mental time travel. So in a healthy state of mind, we may be reminiscing about the past. If we're depressed, we may be ruminating about the past. If we're in a healthy state of mind, we may be planning for the future. But if we're anxious, we may be obsessively worrying about the future. And we and many others have shown that the degree to which these regions are engaged in the form of hyperconnectivity or hyperactivation is associated with more psychopathology across many different disorders. Interestingly, the regions in blue here are showing negative or anti-correlations, and the magnitude of these anti-correlations are related to executive function, like working memory performance, which was first shown by Michelle Hampson, and many others have replicated that and extended it. So we have two main hypotheses. Well, really, it actually boils down to one, that hyperconnectivity of the default mode network is a targetable transdiagnostic mechanism underlying mood and psychotic disorders. And so what we want to do is use real-time fMRI neurofeedback to quiet down these regions that are apparently hijacked in many different clinical disorders. And not only do we see the hyperconnectivity within these self-reference nodes, but we also see that the self-reference nodes kind of rope in, if you will, from a colloquial term, other disease and symptom-specific regions. Like in the case of psychosis or schizophrenia, the default mode network kind of ropes in the superior temporal gyrus, which may imbue self-relevance to auditory hallucinations. In the case of depression, it may rope in the subgenual anterior cingulate, which may be associated with internalization. And this hyperconnectivity, we and others have seen precedes illness, indicating that this may not be a manifestation of the disease, but rather a biological trait, which we can target with real-time neurofeedback early in life. The other form of default network hyperconnectivity is expressed as an underrepresentation of default mode network anti-correlations, which we believe is a transdiagnostic, again, targetable biomarker of cognitive impairment. And so the magnitude of these anti-correlations kind of follow an inverted U-curve. They selectively grow with the executive function in the course of typical development. They peak in young adulthood, and then they trail with aging. And we've seen these significantly reduced in many different clinical populations. And when you're looking at individual fluctuations of attentional issues, you can see that it's predicted by these anti-correlations as well. So there's many different ways to address this. And when I looked into the literature, I saw that actually from more invasive to less invasive interventions, whether it's DBS, ECT, TMS, ketamine, antidepressants, many of them are actually just mitigating this hyperconnectivity in some way. So it would be nice if we could do that with lifestyle choices such as exercise and meditation that can be augmented with real-time neurofeedback in terms of the meditation. So for instance, let's take TMS. Many people target the region of the dorsolateral prefrontal cortex that is maximally anti-correlated to the subgenual ACC, anterior cingulate. And that, although it doesn't work for everybody, is a common technique to treat depression at the moment. And what we found is that this treatment target, the subgenual ACC DLPFC connectivity, actually predicts worsening of anxiety and depression, even in teens who are diagnosed with anxiety and depression, and even in children who are at familiar risk. So if you look at familiar risk population, you can see that the connectivity between the subgenual and DLPFC predicts worsening of anxiety and depression and internalization, and actually predicts conversion to illness. Even in young children who are not pre-selected to be at risk for any kind of mental disorder, this subgenual DLPFC connectivity, the anticorrelations, predict worsening of internalization and anxiety and depression 4 years later. So if we can backtrack all the way to a young normative population, why not try something a little bit softer like mindfulness meditation, where you draw attentional focus to the present moment and metacognitive awareness on ongoing thoughts. So there's a vast literature indicating that mindfulness meditation downregulates the default network, increases GABA, BDNF, and across many different clinical populations, decreases lots of different clinical symptoms. And we and others have shown that it can decrease self-perceived stress in school students and increase sustained attention. Other people have published recent papers showing that the mindfulness-based stress reduction is actually non-inferior to SSRIs to treat anxiety and is relatively similar to CBT for people experiencing depression. So with this information, it seems to me that it would be really excellent to try to get younger and younger kids doing mindfulness meditation. The problem is the very people who might be afflicted with anxiety and depression may not find it easy to do mindfulness meditation, and there's no feedback, so you don't know how you're doing. So this is our current instantiation of real-time feedback. We have our patients practicing meditation. We put them in the scanner, and then we identify their own individual networks, the default network and the frontal control network. As soon as they start doing mindfulness meditation, that down-regulates the default and up-regulates the frontal network. We convert this signal into a game, which is this ball game, and the target, say, is to move the ball up, and the ball moves up when the default is being suppressed lower than the frontal parietal network, and the ball moves with a hop size proportional to the magnitude of the difference between these networks. When the person starts mind wandering and is not doing mindfulness meditation successfully, then the DMN goes up again and the ball goes down. So essentially, the people are moving their mind, using their mind to push the ball up and down when they're oscillating from mindfulness to mind wandering. And we really like this technique because it is non-invasive, and we are identifying the specific intrinsic networks, the functional networks, which is a whole other hour discussion about the importance of that. And it's adaptive. So this ball, if you can't do it at all, the ball hop size will adapt to your ability. And most importantly, I find that this is a network level intervention. So not only are you mitigating the hyperconnectivity within the default network, but you also for free get a mitigation of the hyperconnectivity to this other regions like SCG in the case of schizophrenia, subgenual ACC in the case of depression. So targeting these large scale networks have a lot of different clinical benefits. And this is what I think Michelle was alluding to earlier. And one of the things that I think is best about all of these real-time neurofeedback interventions is that the patients feel agency. Many different treatments, the patients are on the receiving end, whether it's DBS, TMS, whatever. What's really beautiful to watch the transformation in these patients is mindset transformation. Well, rather than being a recipient of an intervention, they're actually the actor in the show, and they see how their brain networks are changing, and they feel how their clinical symptoms are being mitigated. And I have to say there's a lot of excitement amongst the patients. So I'll just give you two examples. One is in schizophrenia. We know schizophrenia is a severe psychiatric disorder that has disturbances of thought, perception, and neurocognitive deficits. And we know hallucinations are a key issue there. And we know SCG is implicated. So we have seen this in many different studies. We see the degree to which MPFC and PCC are correlated predicts outcome. And we see that the degree to which MPFC and SCG are kind of correlated during rest also predicts outcome early before clinical onset. We also know that there's a high comorbidity with anxiety and depression, and that anxiety and depression are risk factors for the onset of illness. And so we can actually see, if you look at clinical high risk, we can see a double dissociation, so such that the MPFC PCC goes with depression and anxiety, whereas the MPFC SCG goes with hallucinations. So here's an example of a patient with psychosis moving the ball up when he's successfully doing mindfulness meditation, and the ball moves down when he's starting to mind wander. And what we see is a mitigation of the MPFC PCC hyperconnectivity goes down, and we see an increase in the MPFC DLPFC anticorrelation. So they don't have anticorrelations at first, but they go down afterwards. And when we try this with a sham condition of the motor cortex, we don't see the modulation of these networks. And importantly, it is the MPFC SCG that most correlated with the reduction of hallucinations. So now we're trying to do neuromodulation in schizophrenia. With targeting rather than the DMN, let's try the SCG. So we tried the SCG, or the superior temporal gyrus, and then found in the real condition, in the real feedback, a mitigation of the MPFC PCC and a mitigation of the MPFC SCG, which were associated with auditory hallucinations reduction. So here I've shown you two different examples where you can target the network and then get reduction in AC and superior temporal gyrus, or you can target superior temporal gyrus and get a reduction in the network. So then you might ask, well, why go for the network? And I think the reason to go for this large scale network is you get other downstream effects that may have relationships to, say, internalization or anxiety and depression. So for instance, we know that there's hyperconnectivity in depression. The DMN kind of ropes in the subgenual. We see this in teens with anxiety and depression, and we see it even in kids who are at familiar risk. So there's hyperconnectivity of the self-reference nodes, subgenual ACC, and the degree to which those are correlated is correlated with internalization and anxiety and depression. So if we have teens with anxiety and depression, we can mitigate that hyperconnectivity both within the network and within the network in the subgenual ACC. And then we also saw an increase in state mindfulness. So we're excited about this because we think that this mindfulness-based neurofeedback may be a transdiagnostic intervention for both mood and psychotic disorders that have downstream clinical benefits. So going forward, we have a number of clinical trials doing this. We're really excited again for early detection and early intervention, as Michelle was saying earlier, to really capitalize on the plasticity of the brain younger and younger. And we also are trying, as Michelle mentioned, to augment DBT that has a component of mindfulness. But we want to scale it. So we're trying to do simultaneous EEG fMRI to capture the DMN and DMN anticorrelations. That's with Aaron Kusi. And then with Clemens, he's trying to passively collect the EEG while doing the real-time feedback in the patients with schizophrenia so that we can scale it up. And then we also know that the GABA anticorrelations tend to go, GABA increases with anticorrelations. And so now with Margaret, we have, we're going to be trying to understand mechanism. So we have MRS, which is voxels are localized with the resting state, and we're going to look at GABA and glutamate pre-post the feedback in clinical high risk. We're just launching off a big clinical trial with Randy Auerbach's team, where we're going to compare just mindfulness alone versus mindfulness feedback. One big question that many people have is, how do you know that the neurofeedback is actually augmenting skill acquisition and utilization? Maybe you're just looking at the effects of mindfulness. Well, we're setting out to actually look at that. Other people have looked at TMS, the cerebellum, and found a reduction of negative symptoms. And so we have another grant where we're looking at the comparison between neurofeedback to mitigate positive symptoms and TMS to mitigate negative symptoms. And with Kristen Webb, we're asking for whom and for when. We know that exercise and neurofeedback both decrease depressive symptoms. And so we're trying to look at the within and between person moderators. I'll just close up now by talking about some of the other opportunities for real-time neurofeedback. Usually people use baseline predictors of outcomes. So for instance, the baseline DMN anticorrelations in our case predict the patient's ability to modulate their networks as well as mitigate symptoms. But the nice thing about the neurofeedback is that we can get neuromodulation predictors. We can actually measure the neuroflexibility or malleability, if you will, based on the change of resting state networks pre-post the day one intervention. And that could be a perturbation prediction. And then when you look at the final change in network, that can be the mechanism. And so you can also think of a kind of an analogy between cardiology and psychiatry, where in cardiology, looking at imaging the heart as an organ of interest during rest and during stress. We're doing the same thing with imaging the brain. during rest and during stress. And you can imagine going to the gym to gain brain fitness. You know that when you're doing exercising, whether it's cardiovascular exercising or pumping iron, you're going to get an increase in fitness. And we can do the same thing with the real-time neurofeedback. So finally, I'll just say that it's really important to identify these within subject fluctuations, and we can do a lot of understanding causal relationships between the brain and the mind, as Tama was talking about, through experience sampling, because then we can actually find out what the person's experiencing. So we've done this within subject prediction fluctuations of attentional issues. We can do the same thing with rumination. And so I'll just finish by showing you one example of where Clemens identified resting state networks, and he did biological triggered experience sampling, where you ask the subject what they're experiencing, and we're triggering the blue areas mean we think they're mind wandering, the subject is responding. So they're clearly in more of a mind wandering state here. Then they can go to the gym and do their mental fitness, and afterwards they're more in a mindful state. And so here, when you do the biological experience sampling, which is much more efficient than experience sampling, you can see that you get a much better mental state. So hopefully we can get the electrophysiological correlates of this DMN and then scale it in the real world, because once we're able to identify optimal and suboptimal brain states, then we can combine it and do digital phenotyping and trigger just-in-time interventions. So I wanted to thank everybody here and Chris for really doing a phenomenal job organizing this, and Michelle has put a lot of effort into this, and all of the program officers and NIH and NIMH in general. Thank you so much. I'm going to now pass it off to Karina Cavito, who's Associate Professor in the Division of Child and Adolescent Psychiatry at the University of Minnesota.
Thank you. Let me start sharing my screen, and then hopefully this will work out. Thank you for your patience. Okay. Actually, Sue teed up to me beautifully, because she actually talked about the importance of taking advantage of the inherent flexibility of pediatric populations. So the topic of my talk today is how to hit a moving target. And
Karina, if I may, we see your presenter view.
Oh, I'm sorry about that. I had that issue before, and I now need to fix it. My apologies. They helped me during the dry thingy, and I can't fix it now. Give me a second here.
Karina, go to display settings again, the drop down.
Give me a second here. Yeah, it's a little bit, so let me just, I'm gonna disconnect from this. And then, okay, so display settings at the top. Sorry about that. Okay.
Yeah, click the drop down arrow under display settings.
So the top?
Yes. And then click the arrow. Nope.
There's something else here, so I can't,
okay.
I apologize. Okay, so is that the one? All right, my apologies. All right, I think I see it now. Okay.
And swap presenter view.
Swap presenter, there you go. Come on, buddy. Thank you for your patience. It's not working. All right, let me do it again. All right, I think I need to project first and then swap. My apologies. I am sorry about the technical difficulties. I need to, yeah. So how about if I stop here for a second? I wonder if Kim would like to go ahead of me so that I can solve this. Is that possible? Kim, do you feel comfortable with that?
No, that's okay. Stop sharing for a second and I will go ahead and share.
Okay, thank you.
Okay. Karina, you may go ahead. Karina, I'm sharing your slides for you, so you may go ahead now with your presentation. Karina, if you're still there, if you come off mute, please.
I'm right here. Sorry about that. I had technical difficulties. So you're sharing the slides from your setup?
Yes, that's correct.
Let's do that then. That's fine. Thank you. Sorry for the delay. So basically, we're going to work today on how to hit a moving target. And essentially, yes, the topic of my my talk has to do, first of all, with essentially what I developed throughout my career. And the first thing that I tried to do was to define and localize sensitive targets in pediatric populations. Then we went on to establish whether the target was engaged and via what processes. And then finally, we are on to the adventure of whether the target is basically linked to behavior or to psychopathology. That we are going to basically do this through 4 different studies that are concatenated and related. We're going to look at a sample of depressed and suicide attempted youth that underwent tasks that had to do with self-processing, and there were 122 of those youth, and then a subsample of those youth that were 81 of them. And then we're going to look into the targets are engaged and via what processes. And that is basically a feasibility study of neurofeedback that we did with very little basically funds, but sort of that brings it to the point that Michelle was trying to make about exploratory studies. And finally, I'm going to give you a preview as to whether the targets are linked to behavior or to psychopathology. And that is a phase innovation study that basically had 50 participants per target. Next slide, please. All right, so I'm going to introduce a very heady concept to all of you, and to the idea that basically self-processing or self-awareness has the basic function of organism survival. And I'm going to orient you to the fact that this very key function has developed both epigenetically and ontogenetically to reach us, and that has endowed us with a capacity for symbolic thinking. And these are the processes that basically allow us for abstract and symbolic representation of the organism. Next slide, please. So basically, symbolic processing relies on the use of images, concepts, abstract relations, language, numeracy, artistic or ritual expression. And only human beings are able to do this, as far as we know. It also allows us to acting contrary to self-preservation, because it allows us to separate the immediate concrete circumstances of the organism from a symbolic representation of the self. Next slide, please. So non-coincidentally, suicide risk changes along development, and it's known that during adolescence, particularly during the ages of about 14 to 20 years, there's an increase in suicide risks. And at the same time, symbolic self-processing consolidates. For the first time in development, individuals are able to represent the self via symbolic, abstract schemas of the self that are separated from concrete self-representation. And one such form of symbolic processing are the self-face images, which, as you all know, adolescents spend numerous hours curating in TikTok and in Facebook and in Snapchat. So really, in some way, social media has turbocharged the ability to extend the self into symbolic ways to both shape who they are and to communicate to others. So I was originally interested in the seat of self-awareness, and by studying suicide, it allowed me to sort of return to my original interest and to the hardcore question of who we are and how do we visualize ourselves and how do we project ourselves into the future. Next slide, please. All right, so with that goal, our lab basically designed this task. It's a very simple task, but is a very complex task. And that is the emotional self-other morph task. And basically adolescents are inside of the scanner and they see around 150 or 200 faces, and they have to recognize the self or the face of a stranger. These faces are also morphed to basically add a certain degree of difficulty to the task. Basically they do this for long periods of time to basically map in the localization and the intensity of activation in brain areas that has to do with self-representation. And it goes really fast. People can do this extraordinarily fast, which is an advantage of the task. Next slide, please. All right, so what we discovered. What we discovered was, and I need to brag a little bit about it, we basically came up with the first biomarker of suicide attempts using precisely this task, self-recognition. Can you hit enter, please? So that you can see then exactly. So what we found was that depressed attempting youth showed higher left amygdala to rostral anterior cingulate cortex connectivity when they are recognizing the self and other faces. And you can see that there's almost a dose response there in terms of how impacted the youth is and how much they engage the amygdala. We specifically interpret this connectivity marker as a marker of compensation and potentially a marker of psychopathological state. Can you please hit return, please? And then I was particularly interested in what was happening in the other connectivity circuit, which was the connectivity between the right amygdala and the subgenual ACC. Hit enter, please. Because what we found was that high ideation youth showed higher right amygdala to rostral anterior cingulate cortex. And interestingly, this did not differ from youth who had strong suicide ideation, but not actual attempts. So I was interested in this marker, and I decided to follow this through either longitudinal or with experimental therapeutics. Next slide, please. So in the meantime, so I'm going to summarize this. We found basically an anterior cingulate to right amygdala circuitry there, and that this was associated with activity and connectivity during self-face recognition. Next, please. So in the meantime, we were also exploring a subset of this sample, 81 adolescents, and we found a very beautiful bilateral cluster in the medial temporal areas, and it included hippocampus, parahippocampus, fusiform, and amygdala. And you can see it very clearly there that the depressed youth that are in green had a lower level of activity in this bilateral cluster when they were recognizing their own self face compared to the control healthy youth who had a very robust activation in this bilateral cluster when they were seeing their own self face. And this is using the same task that we basically use for the suicide attempters. And you can hit enter and you'll see that the task is the same. So when we localized this cluster, we decided to carry it forward and said, okay, let's use bilateral amygdala and hippocampus, provide neurofeedback training to these youth, and see if we can move this cluster in order to change how the individual perceives and symbolizes the self. And that is exactly what we need next. Next slide, please. So basically, our target was the bilateral amygdala and hippocampus. The processes that we were hypothesizing had to do with memory, specifically with the kind of memory that happens very fast, but also very much in reaction to the salient stimuli of the self. And we pair neurofeedback to the self phase in 53 adolescents, controls and depressed. And again, our target was the bilateral and hippocampus cluster and pairing the neurofeedback to the self-ape images. So I'm going to show you how the general outline of the study was in the next slide, please. All right, so they arrived for a first session. They told us their story. We established diagnosis. Then the second session right there in the scanning room, they came up with happy memories. And then inside of the scanner, We provided neurofeedback using the Morphe software, by the way, that Sue introduced to the bilateral amygdala and hippocampus paired to the cell face. Next slide, please. So they also had pre and post tasks. So they completed the same self-recognition task at the baseline. Then they had neurofeedback. Please hit enter. And then they completed the self task afterwards. So let's give you a preview of results as we go into this journey of trying to locate the target. Next slide, please. All right. All right, so this is a zoom in into the task. You can see that the youth were looking at their own faces and trying to upregulate that bar of color that turned green when the target was high, and it would turn red and go down when the target was low. And they were supposed to count backwards to the stimuli of a different face. And the idea here was to engage basically working memory, but also the circuitry that is associated with face recognition, which is very robust and very fast. And this gets to the idea of control freaks. You do what you can and what you can with control conditions with neurofeedback. But this is what we chose as a control condition. Next slide, please. All right. So what we found, unsurprisingly, is that there was a very robust whole brain network that was engaged by the neurofeedback condition, which, as Misaki and others have alluded to, is associated very much with any strong learning task. Can you please hit enter? And then what we noticed is that there was this very interesting engagement of the anterior cingulate cortex for that specific coordinate, which was again reminiscing of what we were seeing with the suicide attempting youth. So we began to say, you know, we need to begin to looking at the anterior cingulate cortex, maybe not as subgenual, but let's hit the dorsal, because there appears to be some kind of cognitive control aspect here, which is very much the function of the dorsal ACC. Next slide, please. All right, so we also found that the youth were in fact able to regulate the amygdala and hippocampus versus the control conditions. And this is, by the way, controlling for multiple other covariates that we enter into this mixed linear model. And that also baseline conditions of the target, specifically amygdala and hippocampus activity during the self-recognition task, was associated with what was happening during the neurofeedback task. So that idea of repeated measures is very much an important aspect of neurofeedback research designed. And we confirm this. Next slide, please. All right, so then we decided to deep dive into this same population, and we looked at the suicide attempters. So in this population of, this small sample of 53 youth, we began to look at the connectivity patterns in the suicide attempters. Next slide, please. And we found, again, a left amygdala story. Please hit enter. As you can see here, we replicated our finding during neurofeedback of a hyperconnectivity of the left amygdala with a number of neocortical areas that were associated with our task. But again, the right amygdala did not differ between the depressed attempted and the not attempting youth. So this was basically a moving target upon which we zoomed in by his absence because it didn't differ between key psychological and pathological conditions. I was particularly interested in it. Please hit enter. So exactly, it wasn't there. It didn't differ between the groups. So I think that we can begin to bring that right amygdala online and see if we can find an interesting curative effect here. Please hit next slide or enter. All right. So finally, we zoomed in into our two moving targets, the right amygdala circuitry, which we know that is associated with fast, implicit, and holistic processes. And the goal was to strengthen fast, implicit, holistic self-preservation, because we know that the act or the actions that lead to suicide in this population are often prompted by impulsive decisions that are not well thought. And then we were interested in the left dorsal anterior cingulate cortex, and our goal was to strengthen cognitive control, flexible attention towards self-preservation. Next slide, please. All right, so that brings us to what we're doing now. And then to the question of what to do, with the time that we have and with the questions that we posed. What targets do we look to? What is important once we have designed this? And this is the FACE Innovations study that I have had the privilege and the effort to conduct. And basically, we put the dorsal ACC and the right amygdala in a race. We made them compete against each other. We provide the youth 2 days of neurofeedback with about 22-minute session. And we decided to say, may the best target win. And we also sample behavioral measures of emotion regulation, self-processing, and psychopathology after neurofeedback. And this is where things get really interesting, right? What is the right outcome to hit your milestones? Are you interested in pathology or are you interested in behavior? Please, next slide. So again, the youth were seeing their own face and elevating when they saw their own face and decreasing where they saw a different face. But basically, this is how the display shows when they're engaged in neurofeedback. Next slide, please. And this is already very much preliminary data. We have concluded our sample collection, and we had an amazing success given the fact that this took place in the middle of COVID. I'm just going to give you a taste of what comes next. If you want to talk more about the timings, I want you to notice that we had follow-ups at around 3 weeks and 5 weeks after the 2 neurofeedback interventions. Next slide, please. And this is what we found. So we have initially very good news. Both targets, the dorsal ACC, which is in black on the left, is associated with significant decrease of suicide ideation over the 3 time points, as well as the amygdala has a significant decrease of suicide ideation over time. There is a very gentle trend, but not entirely insignificant, for the amygdala to be associated with a faster linear decrease of suicide ideation in the youth. Next slide, please. And then again, I pose the question, what do we want to target? Do we want to target psychopathology or do we want to target behavior? Keep in mind that in youth, and particularly in pediatric research, we are often leaping from adult research that sometimes is not unconfirmed or where is not entirely full information in the field. I basically sprung from my colleague Kim Young's research. There's only one publication linked to amygdala neurofeedback in depressed adults. And this was the springboard where I basically reached for my milestones. So I'm going to give you a taste of what we found. Next slide, please. So we basically administered this task that is very interesting. This is basically a dot probe task, and it yields a vigilance score of sustained attention for a given stimuli. And you do that by subtracting reaction times during congruent trials from incongruent trials to different emotional phases. And the phases are negative, neutral, or positive. Next slide, please. And here is the little present that I'm going to leave you with. I had the pleasure of replicating my colleague's Kim's finding, whereby amygdala neurofeedback was associated with increased vigilance to positive phases. And as you can see, when the target was the dorsal ACC, the error bars are very wide, and there doesn't appear to be a strong effect when it comes to attention to positive phases. The other thing that we replicated was the decrease attention to negative phases, but I didn't want to make the talk very long, but we replicated that also. Next slide, please. So I would like to then leave you with this question. I am very honored to be here. Just remember that when you're looking for target engagements in children, you're literally looking for a moving target, a target that is incredibly malleable, full of possibilities, but is based on neurofeedback for adult psychopathology often enough. is often based in psychopathology that is cross-sectional research. However, the excitement that we can offer is precisely the nature of the target's plasticity. And our research has the possibility not only of discovering new brain mechanisms, but also confirming adult findings. Next slide, please. This takes a village. I wouldn't be able to do any of this without my wonderful students, my wonderful collaborators and research assistants. And they will let you know that this was truly a labor of love over 2 years that we were pretty much in lockdown, but we got it done. Thank you very much. Those are just follow-up slides in case that people have questions. I can go back to the follow-up slides to show you. I have a lot of hidden slides to answer questions.
All right, and so it's my turn now. I'm Dr. Kimberly Young from the University of Pittsburgh School of Medicine. And I just want to thank the organizers for putting together this wonderful workshop and for giving me a chance to talk to you all about the opportunities and challenges as we move into efficacy trials and beyond. And so I am going to talk about opportunities and challenges that we have run into with moving fMRI neurofeedback into the clinic. And this is the list of topics I'll be talking about today. And I'm going to start with classification and regulatory challenges. And this is not specific to my neurofeedback protocol. This is specific to neurofeedback, the fMRI neurofeedback community in general. I was specifically asked by several members of the organizing committee and several panel members to discuss this. And these slides on classification and regulation were put together in collaboration with many experts from the neurofeedback community and aren't just my personal opinion. So I think the biggest challenge that's facing us right now as neurofeedback researchers is the issue of where do we belong? And we've touched upon this in the earlier sessions today. Are we a device-based or are we a psychosocial intervention? And currently, all of our applications go to the psychosocial therapeutic and preventive intervention RFA. But there's a question as to whether or not it would be more appropriate if we went to the device-based intervention RFA. And the case for psychosocial is really that we are not actively doing anything to the brain. We're not adding anything or subtracting anything. We're using processes that are already there. Participants are shown activity and they are taught behavioral and mental strategies to learn to control it. The therapeutic effects are induced via learning-based processes. And what's really important and really differentiates us from the device-based community is that the device is not the intervention. The device is augmenting a behavioral intervention, such as savoring, mindfulness, and emotion regulation. Furthermore, all of our studies on clinicaltrials.gov are registered as behavioral interventions, and so moving us to device could lead to some general confusion and inconsistencies. Now, the case for device-based is that we would be evaluated by experts who understand issues that are particularly unique to us related to mechanism, control conditions, and power. The challenge with that is that experts in the device-based community and in the neuroscience community in general do not consider us a part of that intervention. And so putting us together with device-based interventions feels like it's misclassifying us. It's like asking a fish to climb a tree. And this has historically been the case. So in this particular review by Dr. Lisenby, you can see that the definition of non-invasive neuromodulation does not include neurofeedback. It's device-based interventions that apply electrical or magnetic field to modulate neural function. The search terms for the review that were included in this were TMS, TDCS, cathodal and nodal stimulation, as well as electroconvulsive therapy. And here are some more recent literature reviews and meta-analyses on neuromodulation techniques. And again, I'd like to draw your attention to the fact that they do not include neurofeedback. They include TMS, TDCS, ECT, and this graphic right here, again, is based on a 2020 article by Dr. Lisenby that shows all of the devices that are considered device-based neuromodulation. And you can see that fMRI is not included because, again, we're not doing anything actively to the brain. The device is augmenting cognitive strategies. And so this, we're worried that if we go to the device-based study sections, that reviewers would not see us as responsive or fitting in with the RFA, and our applications would not be discussed. And that could lead to reduced funding or even stop funding for novel neurofeedback interventions. And this leads also, being part of the device-based community, leads to the question of whether or not we require FDA approval, which is one of the challenges that I can tell you all fMRI neurofeedback researchers fear. And right now, fMRI neurofeedback is currently very well regulated in the research domain. There's FDA regulations governing scanner usage and IRB oversight of research. There is concern that once it moves into the clinic and it's no longer covered by IRB oversight, it could be used improperly. And that's when we would love to work with the FDA to develop the best regulatory strategies. But we argue, again, that this is more not a device-based intervention. We're more akin to a Fitbit. A Fitbit shows you the number of steps you've taken, and then you can make behavioral or changes to change that number of steps. The watch will even alert you when you have taken too few steps to get you to engage in those strategies. And I argue that fMRI neurofeedback is much more similar to measuring steps, measuring time, than it is to delivering a current to the brain to change neural function. We haven't sought FDA approval in the past because the FDA does not regulate behavioral therapy programs. And we have up until now been very strongly considered a psychosocial therapeutic intervention. And the FDA mission is largely to protect the public health from devices and drugs that present a risk to the health, safety, and welfare of the subject. And there have been numerous fMRI neurofeedback research studies published to date, and there have been absolutely no adverse events reported. So the concern that there would be consumers purchasing neurofeedback equipment without appropriate supervision or training is really not applicable to fMRI, because the MRI scanners are FDA-approved medical devices. And Joe Schmo can't set up an fMRI in his basement and open a neurofeedback clinic. Now, I've been engaging with conversations, my colleagues and I, with the FDA for more than the past decade. And in the last communication we had with them, which was a little bit over a year ago, right before my confirmatory efficacy trial was funded, they cited one of my colleagues had a phone call with the FDA. And they cited a specific title, chapter, and section that said that once an MRI machine is approved for use, if we're using it in an approved way, the specific uses are not regulated. So the function of MRI is to produce images of brain activity using magnets and radio waves, and to see how certain factors affect this function. Neurofeedback is determining how mental strategies affect brain function using stock pulse sequences. We're not doing anything that MRI wasn't originally intended or approved for. The FDA also states they do not regulate the practice of medicine, which is how you use the FDA-approved tool. And that even if we do use the device differently from labeling, it's considered the practice of medicine, which is not regulated. And so one question that I really hope our friends at the FDA answer in the next session is, why now are we being considered device-based, and why now are we being considered as needing FDA approval? It's also unclear what needs approval. So is it the fMRI machine itself? Well, that's an approved medical device. So is it neurofeedback that needs to be approved? If so, is it just neurofeedback in general, and just one of us as researchers need to sacrifice ourselves on the altar of bureaucracy and work with the FDA to get neurofeedback writ large approved? Or is every neurofeedback protocol going to require approval? Currently, if you look at clinicaltrials.gov, there are 95 active neurofeedback protocols. At an estimated time frame of 3 months and $30,000 to get approval, you're looking at 24 years of estimated labor and $3 million that is going to be put onto researchers, not to mention the amount of resources the FDA are going to need to contribute as well. And this might not be a barrier for someone more established, but especially for junior investigators who don't have the same amount of resources as those of us who are more established. This could kill the future of neurofeedback trials. The other question that we could be regulating is software. And as you've heard from the first session, open software that's open source and flexible is critical to neurofeedback development. We are constantly changing our software, constantly improving our software, and that is one of the keys to the advancement of neurofeedback. And the FDA says that software functions are exempt if they're intended to log, record, track, evaluate, or make decisions related to developing and maintaining general health or wellness. We argue that the neurofeedback software allows individuals to track their brain activity in order to make behavioral and cognitive adjustments. There's also the question as to whether or not the software should be considered a medical device, but the FDA defines this as software that is to be used for one or more medical purposes that performs these purposes without being part of a hardware medical device. Neurofeedback software can't be used without the hardware medical device of an MRI machine. Furthermore, it's unclear when we need approval. If, for example, software is what the neurofeedback software we're using is what needs approval, the FDA said that if it meets the definition of a device, but poses minimal risk to patients and consumers, they will not expect manufacturers to submit pre-market review approval. And again, there have been no adverse events reported with real-time fMRI neurofeedback, suggesting that this is a very low-risk device. Also, there's the question of whether or not we would need an IDE exemptions. And the FDA specifies that MRI specifically is a non-significant risk device because there is no potential for serious risk to the health, safety, or welfare of the subject. Furthermore, they specifically state in their regulatory documents that if the IRB determines a study is a non-significant risk device, the IRB may approve that study without the submission of an IDE application to the FDA. So this suggests that if we do require approval, we don't need it pre-market. The FDA did just approve one of the first neurofeedback interventions for PTSD. It's EEG-based, and it was approved in July of 2023. And you'll hear from Dr. Hendler in the next session, but she started out doing real-time fMRI neurofeedback and did not get FDA approval. And she did all of these preliminary trials on the EEG neurofeedback intervention without FDA approval. It was only once she was ready for commercialization and to start her own company that she sought FDA approval. And so we argue that it's premature to seek approval when confirmatory efficacy trials are ongoing, and we don't know if the intervention will go forward as currently designed. So that was just my discussion of, I think, the most important challenge facing us. Now I'm going to talk about some other opportunities related specifically to our real-time amygdala neurofeedback intervention. And one potential challenge, an opportunity that we had, was dose determination. And this is critical for neurofeedback researchers, as the proposed treatment parameters, including dose, have to be included in applications for funding, including R61s. And traditionally, the way to measure dose response has been to deliver the intervention, see the clinical change, deliver another dose, see if the clinical change, and continue administering the intervention until the desired clinical change is reached. Now, as Michelle alluded to earlier in her talk, one of the exciting things about neurofeedback is that the clinical improvements grow in the weeks following completion of the intervention. So here in this 2018 paper, you can see that 2 months after the final neurofeedback intervention, clinical symptoms continued to improve. We have seen this in our own study. And so these are the most current results from our R33 looking at amygdala neurofeedback in treatment-resistant depression. And what you can see is that at 12 weeks follow-up, you are seeing significant improvements still in the active group. And indeed, if we just looked at clinical change after the first two interventions, we would not conclude that the control intervention was, or that the active intervention was any more effective than the control intervention. It's only once we get out to these extended time periods where we see that the individuals in the active group are continuing to benefit, continuing to improve, whereas those in the control condition are not getting as better. And indeed, they're not dropping into below the moderately severe depressed range. So optimizing the number of sessions by embedding assessments assumes that symptom improvement between the adjacent sessions is driven by the neurofeedback rather than the delayed effects of earlier sessions. So this should really inform our neurofeedback designs and suggest that crossover designs may be contaminated by significant carryover effects, as there's no assurance that symptoms will stabilize from the active intervention, even when the arms are spaced weeks apart. So instead, I propose that we should be looking at neural target engagement, not whether or not the symptoms are changing or how much the symptoms are changing, but whether or not the brain, that region that's being targeted, is changing, and whether or not there's a limit to that change. So in our R61 with treatment-resistant subjects, we provided 5 sessions of real-time fMRI neurofeedback. And what you can see when you look at the performance on neurofeedback across visits is that there's a very nice learning curve that occurs on the first day. Afterwards, not so much. And indeed, after the second session, the only blocks that are significantly different from that initial baseline are the baseline and transfer blocks where no neurofeedback information is provided. It suggests that after the learning occurred, feedback actually interfered with learning and performance more than enhancing it. And this is true in studies of other domains of feedback learning, that once you learned what you're being taught, that the feedback will actually interfere with further learning. We can also look at neurofeedback success, which is traditionally defined as the difference between baseline and transfer. Instead, we looked at the difference every day. So where did they start and where did they end on each session? And what you can see is, again, most of the change occurred during that first neurofeedback visit. And by the 5th neurofeedback visit, the change is negative, again, suggesting that too much neurofeedback is interfering with learning. Finally, we can look at when the amygdala activity asymptotes. And so when looking at that information, what we see is that the amygdala seems to asymptote at visit 2. So based on all of these data that show that we really see learning effects in the first visit, and then by the 5th visit, the feedback does not seem to be helping, we have decided that 2 visits is what we are going forward with in our clinical trials. 2 visits are sufficient to engage in learning and to result in the neural changes that we need to see for the clinical improvement to occur. Another potential challenge, but also an opportunity, is predicting individual responses. Because as we know, not everyone is able to learn how to regulate their brain. And so this right here is a combination of the experimental group participants from 3 different clinical trials of amygdala neurofeedback. And what you can see is that about 23% had a neurofeedback success value of 0 or less, meaning that they did not learn to increase their amygdala response with training. We collected demographic and clinical characteristics, the same characteristics of all 3 samples, and then combined them to look at what was related to neurofeedback success. And I just want to draw your attention here to the symptom change and show that we have repeatedly shown very large decreases in symptoms with almost a 50% average decrease with the active intervention. So we threw everything in together and said, okay, what's correlated with neurofeedback success? And we only found 2 variables that were correlated. The first is sex, and our females were more successful than our males. But the second was baseline amygdala activity. And this was how active your amygdala was during positive memory recall before the neurofeedback training. And this suggests that the less you're bringing your amygdala online prior to training, the more successful you are at bringing it online during the training. And when we throw all of these variables into a regression model, the best model that explains the most amount of variance only includes that baseline amygdala activity. And so just to touch on the sex differences for a moment, we do in fact see that females are more successful than males when it comes to regulating their amygdala. But when we look at the baseline, what you can see is that the females have this negative amygdala response and the males do not have, suggesting that it's not necessarily the male-female difference, it's the baseline amygdala activity difference. And indeed, there's a very strong correlation between the baseline amygdala activity and neurofeedback success. And this suggests that our intervention that's targeting a specific deficit requires individuals to have that deficit. And this also suggests that we could do free neurofeedback scans to see if the individual has the deficit and would benefit from the training. Finally, in my last minute here, I'm just going to talk about implementation challenges, or how can we move into part of the clinical world. And one topic that always comes up, and I saw it earlier in the chat, is a note on cost. It is always declared that fMRI neurofeedback is prohibitively expensive. But I want to draw your attention to comparisons with other standard and novel interventions. This table shows a course of treatment. So 2 neurofeedback sessions is a course of neurofeedback treatment. 16 sessions is a course of CBT. And what you can see is that both with conventional interventions and novel interventions like TMS or ketamine, a course of real-time fMRI neurofeedback is not any more expensive than these interventions. And so stating cost as a barrier is not necessarily the reality of the situation. Now, there are other barriers to the clinical implementation. And this is a conversation we're currently working with an implementation scientist, Dr. Kelsey Dixon at San Diego State University, where we're going to be talking to people who are in positions to help real-time fMRI neurofeedback become a clinical reality and ask them what barriers and issues they face that we can begin to address as researchers. And our planned focus groups include MR center directors, MR technologists, as well as insurance company representatives. So just to sum up, we face a lot of challenges and opportunities. There are regulatory challenges. We have, up until today, been considered behavioral and not a device-based intervention because, again, we're not actively doing anything to the brain with a device. And the FDA does not regulate behavioral interventions or the practice of medicine. We've had no adverse events reported with fMRI neurofeedback, and this suggests that FDA approval is not needed, especially pre-market approval. When looking at dose determination, rather than considering clinical improvement, what we should be looking for is target asymptote. And when we're looking at who is going to respond to neurofeedback, in our case, our deficit targeting intervention is only effective in those who present with the deficit. Also suggests that men with depression might have different mechanisms underlying their depression that require different interventions. And finally, implementation is an ongoing conversation that we are excited to engage in. I just want to thank all of the members of my lab and all of the funding that I've received from NIMH and all of you for listening. And now I will turn it back over to Alex.
Thank you, Kim, Karina, and Susan for these great presentations. I wish we could talk about these all day. Unfortunately, we've got until 1:30. I want to start with a question that recently just popped up in the chat. Where did it go? Dr. Young makes a compelling case for unique aspects of fMRI-based neurofeedback that make it a difficult fit for FDA regulation, at least at this time. Earlier speakers discussed the trade-off between exploratory studies to help develop the technology in the field and really focus late-stage clinical interventions. How would additional regulatory requirements for all fMRI-based neurofeedback impact the exploratory type of research that is so critical to the continued advancement of the field?
I think that's a great point, and that is one of the concerns that we have as neurofeedback researchers. It's already difficult to set up and get neurofeedback research funded. If we start having to deal with issues of regulation, especially pre-market regulation, you are going to lose a lot of younger investigators who won't have the resources to devote to that. And again, with exploratory studies, the goal is not to determine that this intervention is what is the final say, is what we're going to go into the clinic with. It's to help us explore. And in putting those regulations on us, I think would really stifle neurofeedback exploratory research.
And before our session continues to answer, I also want to invite Doi Kumsa to join our conversation. She's going to be presenting in session 3, and I think this would also be a good place for her to jump in and welcome her in with us.
Okay, thank you. Yes, we will. I am from the FDA. We will be giving our presentation later on, so we will add more information about what you had asked, Kim. But I think there's some confusion about the regulatory pathway and the things that are available. So we do have a definition for device. So we have a regulation for a rule for device. So it could be an instrument, apparatus, machine, implant, software, including any component, part, or accessory. And if it's intended for use in the diagnosis of a disease or other conditions, or in the cure, mitigation, treatment, or prevention of disease, it could be a device. So I'll put the regulation in the chat so that people can also see that. And I also want to highlight that something could be significant risk or non-significant risk, but that does not mean it will not need marketing authorization to be on trial. So significant risk means you'll have to come in and do an investigational device exemption. But if it's not significant risk, it doesn't mean you don't have to come in for an IDE, but that is a separate situation to coming in for a marketing authorization. So an IDE is not a marketing authorization. And a significant risk determination at the beginning lies between the sponsor or investigator and the IRB, and they determine if it's significant risk or not. And sometimes the IRB could say, go talk to the FDA. So at the end of the chain, in terms of like determining what is significant risk, it does end up with us. But we leave it up to the IRBs to determine if something is significant risk or not. So I hope that's clear. Okay. And
Yeah,
more about the regulatory pathways that are available, indications, if it's targeting, if it's treating a specific indication, it will likely have to come in. So I will go more into those, but I just wanted to give, some highlights.
Thank you. And, that's, again, it's just providing us with some guidelines, because do we need pre-market approval? Do we need IDAs? We just don't know really right now. And we've been told for the past 10 years that we don't.
Right. And we have mechanisms to actually give you written feedback as well. So like there are mechanisms where you come in and ask us questions, do we belong? You can even submit a study, a risk determination. You can ask us questions about, what kind of study do I need? Is my device even regulated? There are mechanisms for that, which I will go into, but yeah.
And just to give a preview, I do hope that everyone will stay for the presentations during session 3, which will be after the break, during which we'll go much more in depth into FDA regulations concerning research, as well as FDA regulations concerning the approval of devices for the treatment or diagnosis of specific conditions, as Doi was referring to. I do just want to clarify that for research, if it's non-significant risk, then an investigational device exemption is not needed. And everything that has been presented today was non-significant risk. And so this doesn't slow down research, but we are using this opportunity to clarify that these are actually devices. And we hope that they will have clinical value and benefit and have a pathway to get into the clinic. And oftentimes the FDA plays an important role in that translational pipeline to get such promising devices into the clinic and get them available for patients. But I'd like to focus this to save further questions about the regulatory status to session 3, so that we can also delve deeper into the really important science and results that were presented by the other presenters here.
If there are no objections to that, we do have plenty of questions to get into in our remaining time here for session 2. One I would like to get into is actually based on something Luke Stokel put in the chat for us, sort of relating to both, I think, what Susan and Karina talked about. I know, Karina, you had mentioned that the translation of protocols across different age groups. So Luke posted a question about sort of the content of your area of work, Susan. He posted a link to a study combining mindfulness, meditation, and exercise in older adults from UCSD and Washington University related to dementia and cognitive decline, but it seemed like they didn't really get positive results from that. So how could, or perhaps you could tell us a little bit more about the process that either Karina may have went into translating a protocol to a different age group, or that may sort of apply to translating some of these paradigms to work with older adults, because that seems like that's a population we didn't necessarily cover that thoroughly here today.
So I would strongly advise, and I hope that you notice that I only presented 3 studies. I would strongly advise, get your preliminarily preclinical data clear, right? What works for this population with this specific developmental history and this at this stage of development. So in terms of translating upwards in development, it would be really important to have preclinical target identification in older adults. And I know that they are at risk, for example, for suicide, especially when they have physical and mental comorbidities that put them at high risk. And then once that takes place, then it's really important to then do a small feasibility study, and then you can open up to exploration. Because let's have a degree of humility here. We're essentially studying the most complex structure in the universe, as far as we know. There's no more complex structure than this one. And there are well known developmental changes that happen well into senectude, and they're all not bad. For example, emotion regulations gets better as individuals get older, because those preponderant impulses that have to do with sexual fitness or even fight for territory and basically jostling with your conspecifics, they become less preponderant when we are already established members of our basically group. So I would say target critical developmental tasks for the age range in question, and identify your target via empirical research. So I recently saw some really interesting work targeting the dorsal anterior cingulate cortex in adults, in older adults. And it was very preliminary and it wasn't very well written, but it was very enticing to look it up. So that would be my general recommendation, to extend into older populations.
I'll ask something else from the chat that sort of relates to a common question we get around fMRI-based neurofeedback. It was specifically coming from your presentation, Karina, but I think this is something that really applies to this work broadly. Somebody asked about how you could validate your work with EEG, and I'll open that up to all 3 of our presenters, since that seems to be a common question that all of our neurofeedback researchers get.
So yeah, that's a great question. And that is one direction that the field is moving forward with, as Sue mentioned, and Dr. Hendler will be talking about, is if we can do concurrent EEG fMRI, can we then use machine learning to find these signatures? Because EEG can't get at the amygdala. And so can we get at, one of these signatures of amygdala activity and have people regulate those as a proxy? Dr. Hendler has been very successful in that. Our lab has attempted to create our own EEG signature based on amygdala upregulation and have not been as successful as her group. But it is something that we are all considering is how we could move this into EEG.
And just to follow up on what Kim was saying, it's not surprising that you're not getting it immediately. I mean, Thomas has fantastic success. It was really a tour de force, but it's really complicated. And this goes back to some of the challenges we've been talking about. The simultaneous EEG fMRI, it's been around for many, many years, and the orders that the the artifacts that you get in the EEG signal are orders of magnitude larger than the signal he's trying to look at. And it's very, very difficult to do this. But we have a few projects where we're trying to capture the electrophysiological correlates of the default mode network. So rather than a region, we're trying to get the correlate of a network, the default mode network or its anti-correlations, which is complicated. But another thing to do is look at just passively collect the EEG and just to see what's changing when you're doing real-time neurofeedback. In the case of schizophrenia, that's what Clemens is doing. But the other thing that is really helpful is to possibly use EEG and or fMRI to trigger on brain states. And if you're using EEG in that modality, you have the higher temporal resolution that you can capitalize on. So there's a lot of really exciting ways both to understand and elucidate mechanism and also to scale the intervention with EEG and fMRI.
Someone else in the chat asked about the validation across cultures. So I was wondering if any of you had any data about cultural differences in neurofeedback response.
So that's a really interesting topic. I suspect that there isn't a lot about that, but I can tell you because of my area of study that the self is a construct. The way in which we visualize ourselves is very much content dependent in the basically the signals that our culture gives us regards to how we perceive ourselves. So we know, for example, that more cultures that are more focused toward fitting the individual in the community have a larger overlap in the areas of the brain that have to do with your own face and the areas of the brain that have to do with your conspecific, so with people of your same community. Whereas in more individualistic cultures, there's much more separation between the self and the others. So there is likely to be a cultural component, not just with regards to something as, as global as our self-representation, but also with regards to how a culture shapes the way in which our brain develops. So there are cultures that highly value basically very tight control of emotion regulation because they're basically they don't have a lot of space. And so getting along with your neighbors, being extremely polite, very much fitting into your community is highly valued and rewarded. So there are other cultures like mine, which are very, very extemporaneous and very, and it's not because we also have no space, but it has to do with our cultural history, because we come from Spain and we're loud and we're expressive and we talk with our hands. And all of those things do have foundational roots in your brain. So I think that is a fantastic question to begin to look at, and it speaks to having a higher representation of diverse communities and diverse experiences in neuroscience and neurofeedback specifically.
I also think it points to the importance of collaboration, international collaboration. We have so many wonderful neurofeedback researchers that are in Europe and Asia. And so combining our data to then look at whether or not there are cultural differences, I think would be a very powerful direction.
Completely agree with that, Kim. Having cross-cultural research in different, in different countries that get at specific processes that vary culturally, that's the way to go.
Hopefully, well, this may be the last question we have time for before we break for 1:30. So I'll introduce it. I've gotten a couple of questions and comments about insurance reimbursement, particularly how the FDA may be involved with that, CPT codes. So maybe starting with Kim and then going to the others, how would we get insurance to start reimbursing for fMRI neurofeedback?
So that is one of the specific aims of my confirmatory R01 is we are specifically engaging in focus groups with insurance company representatives to ask them what they need from us, including whether or not they need from us FDA approval. So that's, I don't know the answer to that. And that's something I really hope that more people will chime in on, and especially in the next section.
I wanted, Kim, do you mind repeating what you just said just now, just in case I can provide
It's just that we are talking to insurance company representatives. We have focus groups planned with them, so we can ask them specifically what they need from us as researchers to be able to approve this and fund this, and whether or not FDA approval is part of that. That's one of our questions.
Oh, gotcha. Okay. It will likely depend on the indication. So for instance, if you're specifically targeting a psychiatric indication, let's say, you're treating major depressive disorder, to market that, it will likely need an FDA approval. Yeah.
So this also just quickly brings up the point of the RDoC approach and the transdiagnostic nature of a lot of our symptoms. And as Sue has, pointed out so eloquently, she's targeting the same region, but for many different disorders, and that it's not necessarily disorder specific. My research has been, but a lot of neurofeedback research has not been disorder specific and does apply to a wide range of mental health issues.
It could be. So as long as it is trying to go into market, if it is targeting a specific disease and treating it or diagnosing it, it will have to come in for clearance or grants.
Well, I am sure this will not be the last we hear of in a conversation about fMRI, FDA, and insurance and other third-party payers. I encourage everyone to stick around for session 3. I'm sure this will come up again. In the meantime, that concludes session 2. Susan, Karina, Kim, thank you so much for your presentations. Tremendously informative, and I really enjoyed getting to be a part of the discussion. For now, it's 1:30, so we have a 30-minute break on our agenda for lunch. So we will resume at 2 o'clock. Thank you very much for an exciting first 2 sessions.